Yeah,
Live. Okay, start. That's what one. The second one. आप चालू कीजिए.
a very good morning to all of you. I welcome you all in the national webinar of application of biotechnological tools in crop improvement. So the webinar is organized basically for the purpose of introduction of recent biotechnological tools to our agricultural student as well as the research professionals of the crop improvement background. Sir, biotechnological tool is basically used for application of desirable gene to transfer in particular crop and identification of that genes. So the purpose of that national webinar is to benefit the student for biotechnological tools like sequencing or molecular breeding or genomics, etc., to apply these tools for crop improvement purpose. In that series, I will request our Honorable Vice Chancellor, sir, and Vice Chancellor Dr. S.K. Rao, sir, and our Director Research Services, Dr. M.P. Jain, sir, for the welcome address, and for keynote address, I will welcome Dr. Nagendra Kumar Singh, sir, National Professor and Director ICR and IPB in the inaugural session. So the session is going to start. So first of all, I will request our DRS, Dr. M.P. Jain, sir, to welcome address.
Uh, Respected Honorable Vice Chancellor of our University, Rajmata Vijayaraje Scindia Krishi Vidyalaya Gwalior, all the dignitaries, participating scientists and students. As you all know, that with the occurrence of COVID-19 pandemic, which is emerging as the most crucial global health problem in of the century, and almost all the nations are struggling to slow down the transmission of this disease by testing and treating patients, restricting large gatherings, maintaining complete or partial lockdown, and restricting travels. But these are the problems which we are facing. But these problems has also led to the creation of new opportunities also. In these crucial times, the scientists of biotechnology, Department of Rajmata Vijayaraje Scindia Krishi Vidyalaya Gwalior, has taken up the initiative to create awareness uh, by organizing this national level webinar series lectures on application of biotechnological tools in crop improvement for the 2 days that is on 10th and 11th June 2020. The webinar is itself is the first in the country, which includes a series of lectures of the prominent and experienced scientists with who have the wide experience in the field of the crop improvement. So this webinar series lectures will cover molecular breeding advancement and approaches, advanced methods of plant phenotyping, genome sequencing prospects, and omics for crop improvements in different scientific sessions. Uh, on behalf of Rajmata Vidyalaya India Gwalior, and on my own behalf, I'll welcome our chairman of the webinar, Honorable Vice Chancellor of RVSKVV Gwalior, who have taken so many new initiatives in the university, including guiding in conduction of these web series lectures. I also welcome our chief guest, Dr. N.K. Singh, National Professor and Director, NIPB, New Delhi, to accept our invitation in short notice and giving his valuable time for this seminar. I welcome all speakers to deliver online lecture for benefits of the students, scientists, and professionals, including Dr. P.M. Gore, Director, Asian Research Program, ICRIS at Hyderabad, Dr. C. Tara Satyavati, Project Coordinator, ACRIP on Parla Millet, Jodhpur, Dr. Sarat Tiwari, Head, Genetics and Plant Breeding Department, JNKV Jabalpur, Dr. A.S.R. Malu, Farmer Dean, Maharana Pratap, University of Agriculture and Technology, Udaipur, Dr. P. Maheswari, Professor and Head, Division of Crop Sciences, VIDA, Hyderabad. Dr. C. Bharadwaj, Principal Scientist, Division of Genetics, IRI, New Delhi. Dr. Rakesh K. Srivastava, Principal Scientist, IFISET, Hyderabad. Dr. Amitabh Mitra, Scientist, NIPV, New Delhi. And I also welcome all the participants to join the webinar and take the advantages from all the scientific sessions. Thank you very much.
Now I will request our keynote for keynote address. I will request Dr. N.K. Singh sir to give enlighten about the webinar and guide us. Dr. N.K. Singh sir.
Thank you very much. Very good morning to all of you. Respected Vice Chancellor and Chairman of the session, Professor S.K. Rao, Dean, Director Research Services of the RBS KBB, Dr. M.P. Jain, and all the other special invitees and designatory who have come here to deliver their special lectures in this one of the first kind of webinar on the applications of biotechnological tools in crop improvement. And I also welcome all the students and other scientific professionals who have joined this particular webinar. And let me say at the outset, I am very happy to be part of this webinar, which has been actually organized actively by none other than our own student, Dr. Sushma Tiwari, who was trained here at our institute and at IRI. So it's a really very welcome initiative at at this difficult time when we are limited by our presence in large gathering and we cannot organize meetings face to face. But nonetheless, this mode is also very useful and we are discovering new things. Um I have later on a detailed presentation about the genomics and its applications, the kind of work which we have been doing at IPB. Here, very briefly, I would like to just tell a few points about the how these biotechnological tools are revolutionizing the crop improvement scenario. Uh, I think all the experts and knowledgeable people, they have the lifetime experience in this area, and they know all these things. Particularly students, I would like to address in this. Uh, that see biotechnology, you know, the definition is the application of the knowledge of biology for the improvement of the living organisms or processes which can be used for the benefit of the mankind. And this industry has grown very rapidly. In the beginning, it was mainly the tissue culture derived techniques which were used for the mass propagation and also for this somatic hybridization, somaclonal variation. These were the techniques which were the first set of techniques which came maybe some about 30 years back. But now it has come a long way, especially the advent of this recombinant DNA technology and our ability to move the genes across the species barrier. That's how it has made different from the other conventional tools, which are limited by our ability to transfer the genes within the species, or at the most in the primary input by classing the closely related species. And those classes were also made with great difficulties, although it has its own potential. So now, as you know, these rDNA techniques were discovered in 1973. First time it was possible to recombine the DNA and transfer. and the first plants were was transformed in 1983. After that, it took nearly 10 years. So in 1995, the first genetically modified organisms were released for commercial cultivation. And ever since, these GMO or genetically modified organisms, which is basically uh, developed by the transformation of the cell with purified DNA, it has taken up, in a big way. And many companies have emerged, even public sector institutions. Many transgenic crops or GMO crops have been released for commercial cultivation. Within India, as you know, that the only transgenic crop or GMO is the BT cotton, which was released in 2003. And after that, different versions of there are some 5, 6 events are there of the BT cotton hybrids. This has been really great commercial success, which allowed India to become a cotton exporting country and also the leading producer of the cotton. The profitability has increased, the productivity almost doubled. All those positive aspects are there on the record. But despite that, there is a section of the society, not only in India, even in globally, which are opposed to this kind of modification on several grounds. They have several questions about that. One is that it is unnatural. We are trying to interfere with the laws of nature. Second is that it may not be safe for human consumption or for the environment. And also on the commercial angle, these products are dominated by multinational companies, so we lose our seed sovereignty and we will be dependent on them. So there are some 4 or 5 key issues. Some of them are genuine concerns, and some are due to lack of ignorance. Really, you can see it is really ignorance. So, but these pressure groups are very, very strong, and as a result, we have not been able to release any other GMO crops after the BT cotton. And you all know that the BT brinjal was ready to be released, and it was cleared by GEST, but then again it was stopped due to these pressures. And our neighbor Bangladesh has been benefited by this BT brinjal for the last 4 years. They are growing and harvesting the benefits of that. Similarly, just last year, a year before that, the GM mustard developed by Professor Deepak Pental from Delhi University, that also has met almost the similar fate. It was cleared by the EAC, but finally, there many people started opposing it, and then it did not see the light of the day. But globally, as you know, more than 200 million hectares of the GM crops are being cultivated. So this is one area which is the biotechnology has very, very huge application, and I'm sure that with time it will become accepted as people realize that this is not as dangerous as people think. And at the same time, the techniques have also now refined. Now we are in the age of the genome editing. What we say that in the conventional genetic modification, the integration of the transgene is random. It could occur any part of the genome. And then you also have to have selectable markers. Quite often it is difficult to remove them from the transgenic plants. But the new genome editing technology is the product is very similar to the conventional mutation breeding. In fact, several of these products you cannot distinguish between whether it is derived from the transgenic technology or it has been derived from the mutation. And as a result, several countries have said that there is no need to regulate this because of the very important reason that you cannot regulate it because you cannot tell the difference whether it was created by genetic termination. There is no foreign gene in that, but nonetheless, you have been able to modify the individual bases a very small portion of the gene, or you can knock down about the expression. So these things have now taken up, and now then again there is a challenge that how our regulatory system is going to face that. So there are actions are being taken around that. So this is a very, very important development, and all of us have to be, as a scientist, as a student, we have to understand these things. And I have seen many times that even scientists starts opposing these things. I don't know for what reason. Maybe they don't understand fully, or uh, they don't want to understand. But all the biosafety issues has to be addressed. But as a scientist, I think we have to appreciate the potential of this technology and how it can help increase productivity and production and the quality, disease resistance, all those things. Now, I will take just couple of minutes more on the other half. Uh, the other part of the biotechnological applications, which is the marker assisted breeding. I'm sure most of these speakers here they will be talking about the success stories of that part. It has actually not faced any objections of that kind, and it is actually delivering very, very successfully. Many products have been delivered. So within the Indian national scenario, improved Pusa Basmati was the first one which was released, I think, in 2007 and around that. And after that, more than 25 varieties have been released through All India coordinated trials, which are derived from the marker assisted breeding. And most of them are actually rice because rice was the forerunner in that. But other crops like chickpea, also Dr. Bhardwaj is there. Now the varieties have started coming in wheat, in other other plants. So it is very, very welcome sign. This actually you cannot introduce any foreign DNA, but you can increase the speed of the breeding, efficiency of the breeding. Almost you can reduce the time by 50% using the marker assisted breeding for the foreground and background selection. So these two parallel techniques, and at the same time, also tissue culture also has come a long way, and now we know that particularly sugarcane, banana, and potato, the tissue culture derived planting material has become a very regular features, and big companies are there. We don't even realize that most of the sugarcane now, at the initial stage, is being multiplied by the tissue culture technique. And this planting material came from the tissue culture technique. So, uh, all these biotechnological tools, in addition to this, there are many diagnostic tools, fingerprinting tools in for the quality controls of the material in the seed business or in the other food business. So there are endless applications are there. So I'm not going to take a lot of time. I just want to close it on a note that these the wide range of biotechnological tools are nothing, these are not a new things. This is the advancement of science of our conventional biology and plant breeding. This is all around these improvements have been integrated into the conventional procedures so that our speed and efficiency has increased greatly, and we all have to learn and understand and start using these techniques, and also to convince, to communicate to the society that there is nothing so much dangerous in it and we should go for it. So with this I would like to close my remarks, and once again thank you all for your attention,
Thank you.
and I will be delivering another lecture in the first session. Thank you so much.
Thank you, sir. Thank you for guiding us and giving your valuable comments. Now I will request our Honorable Vice Chancellor, Dr. S.K. Rao, sir, for presidential address.
Good morning to everyone. I'm Dr. L.K. Singh sir, eminent plant biotechnologist, and Dr. P.M. Gaur from ITSEC, and Dr. Bharadwaj, and Amita Mitra, and Dr. my colleague Dr. S.R. Malu, and Rakesh K. Srivastava, and Maheshwari ji, and then Sharat Tiwari and Tara Satyavati. And all of these are the eminent plant breeders having the very good strong background of biotechnology, spend lifelong time in the crop improvement programs. I welcome all of you and other faculty members, those who have registered from RVS, KUV, and other universities. I have seen larger number of students have been registered for this webinar program because they thought it is a very important program to understand beyond plant breeding and genetics in the routine classes what they are being taught. And now, this is the great initiative has been taken by the Sushma and Tripathi, Manoj Tripathi. I thank both of them for taking this great initiative and organizing to the RVS KVV Gwalior and benefiting the large number of scientific community, especially the plant breeders, the upcoming students in this area. Now, we are passing through a very hard time, that is COVID-19 pandemic, and it has created a global health problems and across the globe, and given a great challenge for the medical sciences to save the mankind across the globe. Now, under these stress situations, everybody is in stress, one means or the other, starting from the political leaders to the educators and academicians, because almost all the institutions were closed for a long period of time, across different countries, starting from schools to the universities. Under those stress situations, then we thought that we should understand the things that we could not able to get a time in the normal days, and this is the high time to have this type of seminar for the plant breeding and genetic students and even the faculty members. And due to many travel restrictions and all, this webinar will be the best opportunity for every one of us to learn many things from these eminent experts. Now, in the crucial time, the today's national webinar on these applications of biotechnological tools, applications in crop improvement programs. Really, it will refresh our knowledge for the faculty as well as for the students as well, and so that we can understand the many things what is going on in today's time. This knowledge definitely benefit for the especially for the students, because the we used to teach many things, like 80% of the syllabus is being taught in the genetics and plant breeding and related activities. In the recently, only for the last decade, we have introduced the subject of marker assisted breeding and marker breeding and all these things. Otherwise, it has been a separate discipline of biotechnology in different universities. Now, all these biotechnological tools, now it should be the integral part of the plant breeding and genetics wherever you go to the any university in India and abroad. And these are the compulsory subjects the students should learn it, because this is the integral part of the plant breeding activity for the crop improvement programs. Why it is becoming so important? Because without these tools, you cannot fasten your crop improvement programs. That is the main problem, because this market assisted breeding and molecular breeding and the genomics, if we are having this much of background and then having complete idea of this, coupled with the smart breeding technologies and then fast track breeding, you can definitely go for the improvement of the develop the varieties to the crop improvement program. It is shorter span of time of 5 to 6 years. As you know, the traditional age-old plant breeders developed several varieties. The scientists told about many crops, and all those about more than 6,000 varieties have been developed in this country with the person not having any background of biotechnology. Because in those days, they never thought this science is of that much utility for the plant breeders. About 40, 50 years back, 30 years back. But now they have gone for only simply hybridization program and selecting the best parents among the genetic diversity available, and then combining those desirable genes into a desirable background, and then come up with a variety after 10 to 13 years. This is a long process. The farmers have to wait for 10 to 13 years to get a new variety. Now the farmers' mindset is also changing now, and then they demand the varieties every year. New variety, what next variety you are bringing to the for us for cultivation? Because the technologies have advanced, they have seen the sense of technology and impact of those technologies since 1950-51 to now 2020. If you look at the food production figures, many times it has been increased 5 to 6 times, and in case of pulses it is doubled. In case of horticulture, it is about double the production within the 15 years of time. In this way, even including oil seeds also, and especially cereals, there's great progress of five times, 5 to 6 times. And all these things, how it has come? It has come only through the crop improvement programs. I am not telling only Vedas has done the miracles. The developmental agencies, they have increased the irrigation capacity different states across the country. That is the number one thing. And the same farmers are same. Those segments get those parental property and those farmers, and they are cultivating the same crops. Almost that is not changed much. Not much crop diversification, especially in food security crops. But what makes the difference in increasing the food security of our system and food production of this many 5 to 6 times is the improved varieties? You are all aware. If you compare the varieties being grown in 1950s and look at the varieties being developed through the crop improvement programs in 2020, there has been a tremendous increase in the productivity of the varieties as well, along with the improved varieties and improved technologies. That makes the whole difference. That means technology is the only base. How this technology has come from? This has come from the crop breeders involved in the crop improvement programs and also other associated staff. Because for every plant breeder, you should be a master of every subject. The plant breeder cannot be a breeder, go for hybridization and then go for marker breeding approaches and then marker assisted breeding and develop a variety. You should have a strong balance knowledge of all the disciplines, including pathology, entomology, including economics, in extension and everything, all, including physiology, agronomy. Otherwise, you cannot develop a good variety. You should be all rounder. You should have a basic knowledge of everything that has to be utilized for the development of the improved variety. Only if you're able to develop a successful varieties in the crop improvement program. This our plant breeders in this country have shown this thing, and in many crops, several varieties have been developed having the very high yielding potential, and even without the much background of the biotechnology in those days. Now the things have changed. Even in the academic institutions, also we have to teach this all these things to the plant breeding students. The biotechnological tools are, you cannot separate it out, or the biotechnology department cannot be separated out from the plant breeding and genetics now. Biotechnology alone, the pure hardcore biotechnology, as Dr. Singh said rightly, there are many areas, including the development of the meat products and all. Many thing applications are there, 150 applications of the biotechnology, hardcore biotechnology. We are not interested in that in our system, in agriculture universities, our food security requirements. We are mainly focused on the agricultural and horticultural crops, not in the livestock and other things. The biotechnological application to the greater extent in improving the breed and all. And now, for these things, our students should have a very strong background of these things. Without that, they cannot survive. They cannot deliver in the public sector or private sector. If any plant breeder goes to the private sector, if they join today, they will last within 2 years to come up with a hybrid or a variety. How it is possible? But they have to deliver it. If they don't deliver it, they will kick them out in the third year. So your services are no more required because you are not generating any income to the company. Similarly, in the public sector also now, there is a lot of pressure on the plant breeders in the institutions to develop the improved varieties of high yielding varieties and already existing varieties. Because the farmers are becoming smarter. Every time they demand, they compare with the varieties developed by the private sector with the public institutions. No, no, this is the variety that last year you have given. Now give me the improved variety better than this year in all respects, so that I can get the more profits out of that and more get the you can get the more returns out of that. It can improve his livelihood. That this is the livelihood based technologies, this crop improvement program technologies and everything the farming communities today. And even if you look at this identification of the QTLs and all these things, we see a lot of papers published, thousands of papers available in the literature. And most of the persons, what they do, they go for some identification of the QTLs and file mapping of the QTLs and all. And after that, they forget. 95% of these applications of this biotechnology in terms of the identification of the QTL stage, they stop the business. Very few persons, like the eminent speakers, they are sitting over here and they are continued with these activities. Those who continued with the activities from the identification of the QTLs to the file mapping and utilization and searching for the new genes and tagging and identification and then validation and then utilization in the crop improvement program, those are only become a successful plant breeders. Those can all only those can be a successful plant breeders and they can deliver the product. I tell you, plant breeding with biotechnological tools, if you cannot deliver the products to the farmers, it has no sense. Simply, publications are equally important. I agree. They go for the best publications in science, nature, and tell them, and advancement of the sciences can contribute. But apart from that, that we should come up with a deliverable product for the farmers. Now that is very important. That is the first requirement. Simultaneously, if some good publications comes in the standard journals, it is really contributing to the addition of advancement of the sciences, but the scientific community. And with these words, I thank this to Dr. N.K. Singh and Dr. Gaur and Dr. Malu and Bharadwaj and Tara and all of my colleagues who has accepted the invitation to deliver their lectures today. And I would like to refresh my knowledge. I will be here the whole 2 days here to hear your presentations. Thank you very much, sir, for all of you. Thank you.
Thank you so much.
Thank you very much, sir. It was really very nice to hear from sir here. I want to include one thing. Our Honorable Vice Chancellor, sir, is very much supportive for such activities. Even I was new to this university, although he has always permitted to discuss anything and start anything new to do for the benefit of students as well as researchers. So now we are going to start our technical session. So for the technical session of day one, the chairman is Dr. V.S. Kandalkar sir, who has recently retired from genetics plant breeding department from RVS KVV Gwalior. And reporter is Dr. Ashok Ahuja sir, who is now he is a guest faculty at our department, but he is former ex-chief at RRLM CSIR Jammu, and other reporter is Dr. R.S. Sikarwar sir, scientist in our department genetics and plant breeding Gwalior. So now I'm handing over the session to Dr. V.S. Kandalkar sir.
Hello, good morning to everybody. This is technical session 1, day 1. I am welcoming all the speakers of these sessions. First of all, I request Dr. N.K. Singh, National Professor and Director. First of all, I introduce him uh, with his biodata. Dr. Nagendra Kumar Singh is the Director, National Research Center and Biotechnology, and National Professors under ICR at National Research Center for Plant Biotechnology, Indian Agriculture Research Institute, New Delhi. He is known for his research in the area of plant genomics and biotechnology, particularly for his contribution in the decoding of rice, tomato, and pigeon pea genomes and understanding of wheat seed storage protein, their effects on wheat quality. He did his PhD from the University of Adelaide, Australia, and doctorate from CSIRO Division of Plant Industry. His major research interests are in the area of genome sequencing and annotation of crop plants and microbes important for Indian agriculture and providing breeders for molecular markers assisted breeding. He is associate with the genome sequencing of rice, wheat, pigeon pea, and mango genomes. His most significant contribution was he discovered a novel class of proteins in wheat seed and named it uh, triticin. Triticin is a legume type storage protein present in the wheat seed in small quantity. He can be manipulated to increase the nutritional quality of wheat. His uh, he was the part of many national and international research projects on rice, tomato, and pigeon pea. He led the ICR team of researchers of International Rice Genome Sequencing Project and led the research which got published in the reputed journal Nature. Uh, it was after many decades that any research work got published in the Nature journal from Indian Agriculture Research Institute. He has the awards and recognition, medalist at BSc program, gold medalist MSc program, and CSIRO postdoctoral award, and National Research Fellowships, Queen Elizabeth II Award, Department of Employment, Education and Training, Government of Australia, and National Bioscience Award for Career Development, DBT, Government of India, New Delhi. He is member of Scientific Panel of Crop Science Division. He is also a member of DBT Task Force on Basic Research in Molecular Biology. He is a Vice President, Society of Plant Biochemistry and Biotechnology, New Delhi, India. Fellow of Academy, National Academy of Agriculture Science. He also awarded by the Rafi Ahmed Kidwai Award, ICR India in the 2007. He is also a fellow of the Academy, Indian National Science Academy, 2011, fellow of National Academy, 2011, and he is also a distinguished alumni award, BHU 2012. He also uh, honored by the Norman Borlaug Award, ICR India in 2015. He has published more than 200 research papers in prestigious journals like Nature Science Reports, PNAS, Close Ones, BMC Genomics, TAG, Ubhaitica, and many more. So I am requesting Dr. Singh sir to represent his talk. Do you hear me, sir?
Unmute your mic, sir.
I think the mic is muted.
Yes, it was actually frozen. Thank you so much, Mr. Chairman, for very detailed introduction. Now, can you see my screen? Can you see the screen that I have shared?
Sir, please share the screen.
Yeah, I have shared it. I think it is taking time to share. Now, can you see it?
No problem, sir. Please.
You can see the screen now?
Yes, sir.
Yes,
Okay.
please carry on, sir.
So, once again, I'm very thankful for the organizers to having invited me for this particular webinar. And I'm very pleased to see, I can see that more than 500 participants are watching this thing on the YouTube live, which is very encouraging. Normally, we will not expect that more than 500 people will come to the lectures. So that is the positive part of it. And the topic of my presentation is the decoding of plant genomes and its application in gene discovery and varietal improvement. Now, at the outset, I would like to acknowledge the funding support from the Department of Biotechnology and Indian Council of Agriculture Research on the whatever work I'm going to describe here. And we have a large network of scientists from many institutes and universities all over India, mostly in the crops which I have been working on, the rice and pigeon pea and also some mango, etc. And then large number of my own colleagues from the NIPB. There's no time to read all those things, but I put on the record just to starting up my presentation. The outline, this is the outline of my presentation. I will very briefly describe that why we need to decode the genomes and what has been the role of ICR in NIPB, and then taking it through to application part, as Dr. Rao has already indicated that these basic research have no meaning until it is translated into product and reaches the farmers and the society. So I will give you one of the very large projects which we have been running again through the networking mode. and the products have been released there. And then also search for new genes is very important, which could be a very long, arduous task. The new genes are present there in the our germplasm, in the line races, in the wild relatives, and there's some efforts we are making towards that. So I will be presenting some examples of that. Now, let us see. I think many of you, particularly the experts, they know these things. But for a larger audience of so many 500 plus, first of all, I would like to tell what is the genome. So, genome was defined exactly 100 years back by a German botanist called Hans Winkler. And he defined it as a complete set of genetic information present in a single haploid cell of an organism, including its protoplasm, means even the mitochondrial and chloroplast genome. So you can see that even the DNA was not known at that time, more than 100 years back. Such a complete definition of genome was given at that time by this German botanist, and it is still holds true. This is the definition of genome even today. And the genetic material mostly it is DNA, but sometimes it is RNA also in case of viruses. As you know, the COVID-19 virus, its genome is a RNA of around 30 kilobase. So why do we need to sequence the genome? As it has been told by several people that these techniques, this research is very costly, and sometimes back people also used to say, no, this is kind of ivory tower research and air conditioning research done in the air conditioned rooms, and it costs a lot of money, very costly equipment. So naturally, we have to have justification. Why do we spend so much money in buying these costly equipments and consumables to do such kind of research? Uh, I have 4 points to tell the reasoning for that. The first is they start from very basic, like it is our human curiosity, the quest for the knowledge that where we are coming from, how where we are in this nature, and what is relationship with the other living organisms, and that is the area of evolution and origin. So having the complete genome information of the different organisms, it helps us to answer that question. The genome sequence is also known as the physical map of the genome as opposed to genetic map. Physical map is basically the base pair, nucleotide base pairs which are present from end to end. Every chromosome is a single piece of DNA from one telomere to other telomere. So once we have decoded that thing, and then we can compare the genomes of different species, and we can know the answer that how far back they had the common ancestor, and complete phylogenetic tree can be created of the entire biosphere, just like we have the family tree, tree people have. Similarly, the all living things on the earth, they are related to each other. They have the common origin. It can be clearly seen and visualized through the DNA phylogenetic tree. So that is a very basic understanding. It also has a bearing on the how to utilize these resources and how to conserve the thing. Now, the second part is that having the complete genome information help us to create the high density fingerprint of our germplasm resources. As we know that nowadays, particularly for the less developed countries, they are very rich in biodiversity, and biodiversity is a wealth for any country, and it is value can be attached in under International Biodiversity Treaty. They can claim rights over that and royalty over that if somebody utilizes that thing. But to be able to prove that, if you want to, how will you prove that this particular material belongs to you? To having genome-wide, very high-density fingerprints, you can always trace back that if anybody is using your material, even part of your germplasm, it can be proven that this part has come from my germplasm, and hence uh, you can claim rights under the biodiversity treaty and that. Otherwise, people will take it and and they will biopiracy, as you know, is very common, and you can never prove that thing. So protection of our biodiversity and claiming the right is made possible by this genomic knowledge. The third is that the gene discovery is itself, the very process of gene discovery and finding the better alleles of those genes for different useful agronomic traits is made much easier by having the knowledge of the genomes. It's just like having a GPS map. Nowadays, the people go around with the GPS in their mobile and navigate through different kinds of cities and different. You can find a person in a city and their address is much easier if you have this map GPS. Similarly, if you want to search a gene, then genome information is just like having a GPS and you can go to the genes much quicker. And then the last point is number four is that the plant breeding itself. Plant breeding is the selection of the desirable genotype from a population, a variable population. And then you have to select the right kind of material out of thousands of lines. Then that selection, conventionally it will be visual selection, or you grow them under some field, some condition. But this selection is can be very much accelerated. I think that is the value of the marker assisted breeding. You have several presentations coming up, and this can be speeded up by having DNA markers, marker aided selection, which is called indirect selection. And there you can at the seedling stage itself, you don't have to wait for the mango tree for 5 years to bear fruit. You can just at the seedling stage itself you can select both foreground and background. So you can see that why we need to spend our resources to decode the genomes of the plants or animals or whatever organism for microbes, because having that knowledge not only satisfies our basic curiosity, which has done to the large extent, but it also helps to protect our germplasm resources. It helps accelerate the gene discovery and allele mining, and also the breeding process itself can be aided by these new biotechnological tools. So biotechnology is not a subject, separate subject as such. Many people, I don't know whether why we introduce, MSc in biotechnology and PhD in biotechnology. But these are a set of tools which are basically applicable to any biological disciplines, as told by Dr. Rao, that even in plant pathology, plant physiology, any biological discipline, microbiology, these biotechnological tools are available. This is a multidisciplinary, you can see, program. Now, in this, here I have tried to summarize that the gene discovery and utilization, both of which are aided by the genome availability, it is a long drawn process. It is not a one of time that you can just take the genome and get the gene in couple of years and then make the variety also. No matter how fast you go through conventional breeding, it will probably take about 15 years, 12 to 15 years to develop a variety using a particular trait. Suppose you identify a trait in the germplasm in a particular low yielding germplasm and you want to put it into high yielding varieties by crossing, selection, trials, etc. It will take about say 12 to 15 years. That is almost roughly that ever since we made the first class. By using marker assisted breeding or molecular breeding, different types of molecular breedings are there, of course. People will talk about these things. This period is almost half to about 50%, 5 to 7 years you can have. Still, you cannot make faster than that because at least 2 years will go into the All India coordinated trials. But this is a great help. And also, it is much more directed and more precise. Breeding becomes more precise what you want to introduce. So it is going from the trait discovery from your gene bank germplasm collections um, using high throughput evaluation to identify a lines having a particular trait value. Then you need to gene discovery. For that, you need segregating populations because you discover the gene basically by association. That when this gene is present, that trait is also present. So for that, you need a structured segregating population where both gene and the traits are segregating, and through co-segregation, using the functional genomic tools or genetic tools, you can identify the genes. Then you go back to the population. Once you get the gene, then every gene has hundreds of alleles. In the beginning when Mendel described genetics, then usually he talked about two alleles only, dominant and recessive. But now we know that at molecular level, every genes have hundreds of alleles, and then we can find that which allele is the best allele which can be used by the in your breeding program. This is called combining. So for this, again, we have to go back to the our germplasm resources, do the resequencing, and then find how many alleles are there and which is the better suited. So after having all this information, then you can use the molecular breeding tools, of course, more precisely. Of course, you can also use the tightly linked markers for that, but it becomes more precise when we have the functional marker, which are the gene-based marker and allele-specific markers, so that we can do precisely foreground and background selection. And then we have to have a large breeding population. One of the major limitations of any breeding program is that our breeding population is very small, relatively small. And as a result, you don't get that right combination of the genes because population is not large enough to uh, have any chance of having the best combination of genes. So this way, using these high throughput tools, one can deal with very large population, and at very early stage of the at seedling stage itself, you can practice the selection, and then you can narrow down the population for going multi-location trial, etc. Because eventually the trial has to be done, but then you can compress your breeding selected material and get the best of the best varieties. That's how these tools actually help us. Now let us come back to the genome. I will just give you a very brief overview of this thing. Genome sequencing, of course, we know the genome sequencing techniques. These techniques have evolved over the years, and during the last 20 years, they have seen great revolution in genome sequencing technologies. And I will just give you by example without going into too much detail. The very first thing this slide tries to demonstrate that there is a huge variation in the size of the genome. Particularly, plant genomes have huge variation in size, mostly due to polyploidy and also due to amplification of the transposable element. Most of the genome of the larger plants are animal visit repetitive sequences, which are mostly due to transposition, transposable elements. Different kinds of they are there. So it becomes very, very difficult to sequence the genome because the sheer enormous size of the genome. For example, we see the wheat genome, which is 40 times bigger than the rice genome, which is 17,000 MB, and it has taken more than 10 years and 20 different countries joining hands together, and still we don't have high quality reference genome of wheat. We have a reference genome of wheat, but it still has more than 2% of sequencing gaps are there, the youngs are there. So it's mostly the size which makes the task very, very difficult. And 1 picogram of DNA is 1 billion base pair. This is something to remember. 1 picogram is 10 to the power minus 12 gram, and 1 billion means 10 raised to the power 12 base pairs. So see this kind of the enormous size and complexity of DNA is there. It is such a fine, very fine thread of life, and so much information is packed in there to get that information out and arrange these chromosomes into its original form is a huge task. And it is just like a joining the jigsaw puzzle. When the original picture is very big and pieces are very small, it becomes very, very difficult to join the puzzle. So larger genomes, they become very, very difficult to sequence. And that's why very few, despite so much progress, very few genomes have been sequenced to the reference quality. Uh, Now, India, in India, we entered the genome program in 2000. Before that, if you say the internationally, the Human Genome Project and the Arabidopsis Genome Project as a model animal and plant genomes, they started in 1990s and they nearly finished in 2000. And that's where we started. You know, we were not part of these international efforts, and we keep on comparing with the China, and China was part of the both Arabidopsis as well as the Human Genome Project, and as a result, they have benefited from that. We started in year 2000 with the Rice Genome Project, and I will not go into detail because this was a clone by clone approach. This was done using clone by clone approach because we have to clone the DNA for sequencing. And if the genome is large, then you have to make first bigger back clones, and then again break into pieces and make the plasmid clones, and then sequence, and then reconstruct the back clones sequence, and then at the chromosome level. Now this was released in 2002. In fact, we finished it ahead of the schedule, the draft genome, and then in 2005 the paper was published in Nature, which has been cited more than 2000 times and been used in by geneticists and breeders worldwide. And every year, this sequence information is being used. This is just like a reference material, just like having a map of the world. You use the map. Similarly, for the rice genome, you use this map for different purposes. So India has contributed in that genome sequencing effort by chromosome 11, Delhi University South Campus, and our institute from ICR. We completed that work under a DBT project. Then I just want to skip so many things going into detail. So we start developing the genomic tools which can be used for the application part. So one thing we designed is a single copy gene based chip, and this is the patent is under final stage here. This particular chip has got, you can do 96 samples at a time, and in each bell what you can see here, there is a bell with 12 times 8 samples you can do, and in each bell you can genotype 50,000 SNPs. Now this particular is fabricated by Epimetrics, but the chip is designed by us. That there are 50,000 SNPs. They are mostly coming from the single copy genes, but it also includes the agronomically important genes and other kind of genes. And advantage is that we have multiple SNPs per gene, so you can get the haplotype information. You can get the allele information about those genes. So I will tell about the application later on. After that, we have completed the tomato genome sequence in 2012. This was published, and the entire chromosome 5 of tomato was sequenced by India. And once again, it was the Delhi University South Campus and our institute plus NIPGR, which also did part of that thing. So three institutes together contributed to this international effort. And in these both rice genome publications and the tomato genome, there was a major contribution because more than 25 authors were from India. That shows that importance, how much contributions were made, and then how much learning was there. It's not the contribution and publication. It is the, see, many times people say that publication is not the issue at all, and publication is also not the output. Publication is just the record of what we have done. It is a written record of what you did. Whether you do basic research or whether you release a variety, you can write a good paper on a mega variety you have released, that will be also cited 100 times. So publication is not our achievement. Publication is only record of our achievement. It's a written record of our achievement, documentation of your achievement. So it is mostly we should compare the what knowledge we have generated or what the product we have generated, rather than what we have published and we have product. It is not publication versus product, but it is basic knowledge versus the transnational product. So these sequences being used once again more than 2000 citations, because many tomato scientists use this for developing markers, developing varieties. So it's a useful contribution. Similarly, the wheat genome draft came in Science in 2014, and the reference genome came in 2018. This is what I was talking about. The wheat genome is such a huge size that even with so many countries joining head together, it still requires a lot of work before it will come to the same level as the rice genome. Rice genome is actually gold standard because there are very few yarns and very high quality sequence. In case of wheat genome, it is not like that, because mainly because of the size problem and lot of repetition there. Now let us come to the pigeon pea genome. Now those three genomes which I told you, that rice, tomato, and wheat, they were done through international partnership. But then we learned how to do the genome sequencing during this process. And also one more thing I must tell you, that the initially what was clone by clone sequencing at the time of rice genome, and halfway through the tomato genome in about 2008, these techniques changed actually. There was a next generation sequencing technology, what we call NGS in short. They evolved during that time. So the sequencing became much faster and much cheaper. And there were three techniques, three parallel techniques were there. One was the 454 called pyro sequencing. Then another one was the Illumina sequencing, and then another third one was the solid sequencing. There were three separate techniques, but one thing was common, that you could sequence thousands of large number of DNA fragments in parallel, so you can generate huge amount of data. And then another thing was common is that you don't have to clone the DNA. So the cloning was making the whole process very, very slow, because you have to first clone in the back clone, then again you clone into plasmid, and each clone has to be purified and picked up using a toothpick, and then purify the plasmid, then sequence. Very tedious process. That's why it was slow process and expensive process. Then when the NGS came, then the cloning was not necessary. The cloning was replaced by PCR amplification. So as a result, we were able to, through an Indian network of different universities and we were able to sequence the first draft of the pigeon pea genome sequence in 2011. And this was the first in India also of any eukaryotic genome, and first legumes in the world. And subsequently, Chris had also sequenced the pigeon pea genome and also chickpea. So now this sequencing became, much easier now. It was more now many genomes came, and most of them were draft genome. That is another thing. That is the downside side of it. Very easy to create a draft, but very difficult to create a reference quality genome, because which have the continuous sequence from telomere to telomere with very few gaps and high quality sequence data, pseudomolecules. This is still a challenge. So we still don't have reference genomes of pigeon pea and chickpea and many crops. Even as I told you that in wheat genome we call it a reference genome, but still it is not that high quality reference, because there are 2.5% sequence gaps are there, YANs are there. So in pigeon pea this was done, and once you have the genome sequence, then you start finding applications. Then we have high density map, we can do gene mapping, QTL mapping, and then same thing what has happened in case of rice, this can be replicated in almost every crops. But there will be a time lag will be there. It will probably take 5-10 years for pigeon pea to reach the same stage where rice is now. So we mapped the couple of very, very important loci here on chromosome 4 and chromosome 5, which have genes for the plant type traits, which is the plant height, maturity, branching, and this can be used in breeding. through marker-assisted selection. Of course, many people will say that why we need to do need to use marker for selecting for plant height. That's a very good question, because height you can really see that the plant is dwarf. But when you want to, select for many genes together and at early stage of the plant, at seedling stage, then it could be very useful to have markers for even for simply inherited traits also, because at the early stage you can compress your breeding population, so you can do field trials on a much smaller set of lines which have combined all kinds of good genes. So in pigeon pea still we don't have mass derived varieties, but slowly we are moving towards that, and the dwarf varieties are becoming order of the day also here, and one Pusa Arhat dwarf 16 has been released, and it is showing a lot of promise. More lines are coming up in that direction. So just like I told you, in case of rice, we developed a chip. We have developed a chip which is published this year now, with 62,000 SNPs in 9,600 genes. Now these genes, we have also added the 747 genes for disease resistance genes. So you can see here is the listing of the agronomically important genes, then single copy genes, then disease resistance genes. And in the last column, you can see the number of SNPs per genes. Now this is the uniqueness of the chip which was developed at NIPB, for which the patent has been filed in case of rice. So similar chip we have developed in pigeon pea and also in mango, which I will be telling you very briefly. You can see on average there are 6 SNPs per genes. So that means you will not be only be able to score for the SNP as a marker, but SNPs are bi-allelic in nature. Like for every SNP, there are only 2 alleles. Okay, there are reasons for that. I don't want to go into this. Although theoretically 4 alleles are possible because you have 4 bases, but usually you will find only 2 alleles. But if you have number of SNPs per gene, then you can have haplotypes, and then you can get multi-allelic information that each gene has maybe 10 alleles or 50 alleles, and then which allele was associated with a particular trait value. That means you can do gene-based association mapping, and you can identify the alleles which is responsible for that particular trait. So these are the applications of this resource which have been generated. Of course, now we have started using it, and it is open for anybody to use it, any pigeon pea breeder or geneticist. Now, this was already this paper has been published, where it has been used for the number of grains per pod and branching pattern. Some of the useful agronomic traits it has been achieved, and any trait, fertility restoration or whatever trait is segregating in your mapping population, one can analyze and get very high density map, and then move on from there in the breeding applications. Similarly, in case of jute genome, we published this with more than 90% coverage, the draft of the jute genome. These were done in the Indian situation, and a QTL has been identified. Here is the partnership with our Jute Institute, CRIJF, in Barakpur in Kolkata. With Dr. Sarkar is the main worker there. And the jute fiber quality QTL has been identified and being used in the breeding program. Now let us come to the mango. Here actually we took the crops which are very much important for Indian context. That means they are very high volume, high value, but there will be not much international interest, because be it a jute or pigeon pea or mango, it is mostly grown and consumed in India. So other countries will not have that much of interest to put their resources or their money into these things. So we picked up some of those crops which are important for India but not for the world, and that's where we should invest to generate genomic resources in that. And mango was one of them. It is more than 10 billion US dollar worth product every year, but there is not much international trade, and then 70% or more is produced within India, but it is becoming very, very popular globally. Now, mango, the huge potential for export. India has so much variability. But when we started sequencing the mango genome, we faced a really very big challenge. Even though mango genome is small, same as the rice genome, but it has very high level of heterozygosity. And I think many of the plant genomes will have that problem, where you don't have the pure lines, you don't have the inbred lines, you don't have double applied. Mostly trees will be in that nature. Many of the crops will be also like that. So this heterozygosity creates a problem in genome assembly, because almost all the genomes of plant and animal which has been sequenced till date, they are the haploid genomes. Means we assume that they are haploid. Each chromosome is in a pair, and those pairs are not identical. There are variation, there is heterozygosity. But if you see every genome which has been submitted and published in NCBI, you have just only one thread running, as if there was only one chromosome. And there is no account of the heterozygosity and diploidy. But these genomes like mango, which have very high level of thing, that becomes a very, very challenge. So actually we need to represent each genome by two chromosomes, one pair of chromosome. Each chromosome has in a pair. So how we can distinguish? We can show in only one line. It was done because we mostly sequence the pure lines or double haploid lines. So that was not a challenge. But in case of wheat, so what happened is that when we assemble the genome, the assembly size becomes larger than the genome size. That was the challenge. So we got 492 MB of genome assembly. Genome size is almost 400 MB. And this was because of high heterozygosity. The male, the chromosome, but the paternal chromosome was assembling, at least part of the paternal chromosome will assemble separately than the maternal chromosome. So it will not collapse into one single chromosome. And now, how this problem was solved, to cut the long story short, excuse me, uh, this problem was solved by making use of the latest development in sequencing technology, that is called single molecule sequencing. This single molecule sequencing, they generate very long reads. There are two technology are there presently available. One is the PAC Bio, Article Pacific Biosciences. The other one is the nanopore, Oxford nanopore technology. Both of these technology, they use the DNA and then you can sequence them directly. Now this is called the third generation sequencing, where we don't even need to PCR amplify the DNA. We just isolate the DNA and sequence directly, and you generate very, very large reads of tens of kilobases as compared to only 100 base pair in Illumina, and maybe at the most 600, 700 base pair in the Sanger sequencing or 454 sequencing. Here you can generate reads up to 10 kb, 20 kb, like that. So what is the advantage of having very large reads? The advantage is that then we can assemble, even with the heterozygosity, we can allow for the heterozygosity by allow the mismatches while assembling the DNA. So it will take time to explain the nitty-gritty of that, but you can see that the approach which we followed, that we went for the large overlap and allow for the heterozygosity by allowing mismatches. Then we were able to assemble the mango genome. But that is not a internationally recognized method actually, that we had innovated this thing. Now, meanwhile, these internationally recognized methods came. The software came, like the Falcon and Jeep program came, which somehow separates the haplotics into diploid contigs. And also, one of the another latest approach came, is called the trio binning approach. Trio binning means the two parents and the progeny. This is the trio. And first it was used in the mammals, in buffalo and cattle, and even in some fish. In plants, it has not been used at all, but first time we used it in Amrapali. because we were lucky that both the parents, Neelam and Dasari, and the progeny, which is Amrapali, they are there in the IRI orchard. So having you must have the parents, the two parents, then only you can sequence the progeny. Because the parents DNA, parent sequence is helpful in separating the two chromosomes of the pair, maternal and paternal. So to cut the long story short, because of our ability to separate the maternal and paternal chromosomes, we have been able to assemble the genome of mango in the face of international competition. Because even Australia and USA, they were also involved, but every one of us, we faced this difficulty. The other people did not have the parental lines, so they could not move further. They had to stop after some times. And recently, some Chinese, two Chinese people have also come, but they have the same problem because they don't have the parents, so they cannot assemble accurately. Now, this mango genome work, it is submitted to NCBI 2 years back, but still it has not been published. Actually, it is published in the summary form, but detailed publication is under preparation. But you can see mango has huge variety variation. You can see here more than 1,000 varieties with different shape, size, and taste and color. You know that mango season is coming. It is the king of fruits. So this great variability was analyzed, has been analyzed at the um, morphological level and biochemical level and um, these kinds of level, but not at molecular level. So now with this availability of genome sequence, and we developed a chip for mango also in the very same principle, this time 82,000 SNPs. And we have all the disease resistance genes and horticulturally important genes, single copy genes on the same lines. You can see we have genotype 400 varieties of mango. You can see one allele, the second allele in the middle, the plants which are heterozygote. So you can see there is large amount of heterozygosity is there. Now there are tools are available which can make use of this heterozygosity also, and you can do association mapping. For example, here the fruit acidity. There was a QTL here was identified to genome-wide association mapping. So whether the some varieties of mango are very sour, some are sweet. Even without maturity, some mangoes are sweet. They are not that sour. And there is one major gene we identified on chromosome number 11 with very high large score of 22. Similarly, we have identified genes for pulp percentage on three different chromosomes. That is ratio of the pulp to stone. Then also, I think something got stuck. We have also got the genes for the size of the fruit length and fruit weight, and alternate bearing. A number of things actually we have identified. So this can be used in the mango breeding, particularly in tree breeding, which are long juvenile phase. This kind of genomic resources and marker-assisted breeding will be a great help. So just I gave you just very briefly some flavor of the genome research which were done internationally. Three genomes: rice, wheat, and tomato. And in India, we did pigeon pea, jute, and mango, which we have done, and it has huge potential for application. Now I want to give you some very quickly some examples of application actually. One of the, I think most of the varieties which have been released through marker assisted breeding are actually through backcross breeding program, because we take a highly adapted popular variety and we transfer a gene in that for value addition. It could be for the abiotic stress tolerance, it could be disease resistance, it could be anything. But we take advantage of the already established variety, and that is the principle of backcross breeding. So in the conventional backcross breeding, we have to make some 8-9 backcrosses to recover the background. But now with 2-3 backcrosses also, we can use foreground background selection, and we can recover more than 90% of the genome. And then there is a nil trial. A trials are there where you can have the near isogenic lines, and after 2 years of testing in AVT, A number of varieties of rice has been released, and I will give you some examples of that. So you can see here that what is the challenge of high yielding varieties of rice and wheat particularly. These varieties were bred for the high input condition, and mostly at the early stage the selection was done mostly for the yield and to some extent for the disease resistance. But the abiotic stresses and the quality, these things were not taken into care that much, and as a result, most of these mega varieties of rice, which are very high yielding varieties, they are very popular in the farmers because they are very high yielding and they are adaptable. But traditional varieties of rice, they were very popular earlier on because they had the tolerance to different stresses and they had good quality. But now farmers, they don't like to grow them because of the low yielding. So we have to combine the two things. That means we have to identify the genes for stress tolerance and quality from the traditional variety and transfer them into modern high yielding varieties, which can be done through the backcross breeding. And the backcross breeding can be accelerated by marker assisted backcross. So, this is the scenario here. The many people say that green revolution has done lot of damage to agriculture in India because we lost all our traditional varieties. Many conservative people they tell like this, but they don't understand that why farmers stopped growing those traditional varieties. The single most important reason is that they were very poor yielders for different regions. Even though they had tolerance and they had good quality, all the good things were there, but still all those varieties are there in the gene bank of NBPG. If anybody wants to grow, we can make available the seeds, but farmers will not grow them because they don't yield that much. But for the mega variety, green revolution variety, they yield very high. Farmers also knows that the quality is not as good and it needs a lot of water. But the breeder's job is to combine the two things. So in case of rice, there are many varieties which are called mega variety. They are grown in millions of hectares, but they lack the climate resilience, and many times they don't have good quality. So that's where here I've tried to just summarize that. See, molecular breeding or marker assisted breeding, there are different ways you can do. Like you have the mass pedigree methods are there. Marker assisted pedigree method could be there. Marker assisted recurrent selection could be there. Genomic selection is coming up in a big way. Then you have marker assisted backcross breeding. Out of these four, the marker assisted backcross breeding is the one which is being most successful for several reasons. I will not go into detail because we don't have the time, but mostly it is because of it is a numbers game actually. In the other methods, you have to apply this breeding into larger population, and then to get the right combination of the right assortment of the genes is dependent on the population. Even if the even very few number of genes are segregating, to have a right combination, the population has to be very very large. But in the backcross breeding, already you have a very right combination of the genes, except that it lacks certain certain genes, certain resistance or quality. So here you see one example is there. The Swarna Sabon rice variety. The Swarna variety is very high yielding, very popular for the last 30 years or more, 40 years. But it is sensitive to submergence. If it is submerged in the water even for 2-3 days, it will die completely. So a gene for submergence tolerance was identified in a land race called FR13A, and this gene was discovered through genomics. Genome availability is speeded up the gene discovery through map-based cloning. Like the having the GPS of the genome helped to identify the gene, and then within 3 years the gene was transferred into Swarna variety. So now you have Swarna sub 1. Now this Swarna sub 1 will not die even the plant is submerged for 2 weeks. And you can see here within 3 years or 2.5 years, you can transfer the small segment of the FR13A into Swarna, and now this Swarna sub 1 has all the good quality yield resilience for Swarna, but the added advantage of the submergence tolerance. So this is just one example, and many genes can be transferred just like that. Once you have the information about the gene which is responsible for the trait, and you have the tools to select the backgrounds. Background recovery is very very important because otherwise you will not have the original Swarna. And why original Swarna? Because original Swarna has proven the test of time. For last 40 years, it is one of the ruling variety. So similarly, all the mega varieties are like that. Mega varieties of any crop, they are the rare combination, rare assortment of the genes. And once you cross during the breeding, you disturb that rare combination. So how you get it back? You get it back by back crossing. So there are two different approaches. One is to develop a new variety, to develop a new combination, which may be better than Swarna. There is no denying that, and people are trying to do that. The second is that put in Swarna what it is lacking. It is already good combination. So, and so not only Swarna, say IR64 is there, MTU 1010. So in any other crops, even like in pigeon pea, maybe you have the variety like that. In chickpea also, in wheat also, there are variety which are mega varieties, but they just lack one or two traits which you can add by crossing. So it's a very legitimate method of breeding. Here you can see the sub-1 gene was transferred into many varieties, and they all performed very well because we are doing precise breeding. We are transferring a gene segment, a gene which does particular function, and we are recovering the background. So it's just like a new version of software. You have the software Microsoft Word 2.1, Microsoft Word 10.1, Microsoft Word 14.1. This is all Microsoft Word, but you added features. So improved versions of that. So this chip we use to analyze the background because initially the background selection was done using low density SSR markers. But when we use this high density chip for background analysis, we found that these four varieties which were developed by the ERI basically in Indian collaboration, Swarna sub 1, IR64 sub 1, Savitri sub 1, and Samba Masuri sub 1, you can see the background recovery range from 96% to 83%. That means the background recovery through visual selection, it was not able to identify, even through low density background selection markers, it was not possible to identify the whole genome background recovery. And there was a lot of variation, more than 10-15% variation was still there. And this is reflected in the acceptance of these mass derived variety by the farmers. While Swarna Saban is very well accepted, Samba Masuri Saban is not that much accepted because still 20% of the background is missing. So that means background selection is very very important, but it can be done at the later stage of the breeding because initially you do make a lot of gains in the background. Every generation you make 50% gain in the recurrent parent background, so there is no need. It is wastage to do background selection at the early stage. Better to do at the later stage because once you have this selected a set of lines, and then from there you can find which line has got the maximum recovery using the chip. Now, just giving one more example that this is one gene we discovered for the high grain number in rice using again all the genomic tools on chromosome 4, and then we transferred this particular gene into 12 different background of mega varieties of rice. This all includes Samba Masuri, Swarna, Pusa 1121, MTU 1010, Ranjit. I think the 12 best varieties of India means which is time tested mega varieties. We transferred in all of them this particular gene increase the grain number per panicule. And you can see one of the best effect was in Samba Masuri. You can see more than 100 grain. This is B, the second one. This is Samba Masuri on the left and the Samba Masuri QTL 4.1. You can see the panicle size in almost all background. So this is a very good QTL. Now we are advanced stage of testing. It increases the yield actually 10 to 14% in different depending on the background. And using the same methods of background and foreground selection, we have developed these lines and we are testing them. Okay, here is one example. This is on chromosome 4. This is the QTL, and the blue background is the recipient parent has been recovered, except for in some regions on chromosome 2 and some here on chromosome 5. One more back class will get rid of that. But at least you can visualize that how much background and where it has been recovered using high density fingerprint for background analysis, if not selection. It is also selection because out of 20 lines, we choose one line which has got the maximum recovery. So that is a selection, but at the later stage of the backcross. Now let's come to the third point. Now this was one example of, I showed you the Swarna sub 1 and then with the QTL of high grade number. Now this is the large program funded by the DBT. First phase was for 5 years, then the second phase came for the 3 years. Here the aim is to transfer these QTLs for submergence tolerance, salinity tolerance, and drought tolerance into mega varieties of rice through a network of 15 institutes from India. And these are here mostly in eastern India where the agriculture is rain fed. And then these states becomes very, very important. The huge yield losses are due to drought, flooding, and salinity in there. And there are 7 QTLs for drought tolerance. These are called DTY, means yield under drought. We don't know the genes here, but we know the QTL very narrow segment which controls this yield under drought. And then the sub-1 genes and the salt tolerance genes, the total 9 genes we have taken for transferring. And this approach was published. So this is totally breeding approach. It was published here. And this paper itself is quoted more than 80, 90 times. This was simply describing the approach. After 2, 3 back classes, how will you do background selection, foreground selection, and come up with this variety. So these markers were standardized, polymorphism at NIPB, and then information was passed on to the breeding partners, and they are doing this foreground selection using SSR markers. Then fine mapping was done to break the linkage between SD1 gene and one of the QTLs for drought tolerance was very tightly linked with the dwarfing gene. So we will never know that all our dwarf varieties have have the poor allele. This is called linkage under repulsion. That means one good trait is linked, good allele is linked with one bad allele. So very tightly linked. So all the dwarf variety, they have the sensitive allele of this QTL. And as a result, they are susceptible to the drought. So we were able to break this linkage by high density, by fine mapping. Now we have I can show you here. I'm not going into detail. I know I'm running out of time very quickly. You can see here, this is the left side. It is the original, right side is the original Swarna. It was planted at Raipur, IGKV Raipur. You can see it has almost dried. And this QTL which is recombined, that means it has the dwarfing gene, but this tolerant allele of that QTL, uh, it is actually surviving. It is green still and surviving. That means this unholy linkage or undesirable linkage between a good and a bad allele of agronomic traits can be broken down, and then it can be followed in the breeding program. That you can see using the marker, you can see that which allele has come. Now the second last part is that search for new genes. So the genes and alleles are already present in our germ plot. You see, the challenge is that which I showed earlier is to find all these good genes and alleles from our germ plant resources and put them back into very high yielding background so that you combine yield with the resilience, yield with the quality. This is the whole breeding process is that we have to find the genes from all over the places and then put into one background which have got the best of everything. And for that, you have the knowledge of, as the Honorable Vice Chancellor told, you have to be good. You have to have at least knowledge of physiology, also biochemistry, also breeding, also molecular biology. Also, you don't need to be expert in all, but you have to appreciate that each of these disciplines are very very important. The agronomy also, that what kind of genes are required to grow in a particular area, and also social science is most important, that what people want. It's not that what we are giving them, that take this, take this, take this. What they want, first you ask what you want and then breed that what they want. So all this integrated solution has to be provided. And then the gene discovery, I just giving one example, that even though we have the nearly more than 100,000 land races and traditional varieties stocked up in the NBPGR gene bank, the wild rice, which grows very widely in the habitats, natural habitats, India is one of the center of origin, was not collected. I started this program about 10 years back when there were only 200 wild rice successions were there, although now they have reached about 7 800 now. And I have also collected around 1,000, because it's very difficult to collect and maintain. So this program under National Professional Scheme, it was due to collect these wild rice resources from different parts of the country, which are growing under the hottest spot. This is the wild rice. You can see this is black color. It's got very long arms, and it has got this shattering property because it has uneven maturity and it shatters. So it is quite difficult to collect and also maintain. And I collected from all parts of the country, from out of the 15 agroclimatic zones of India. I have collected from 10 agroclimatic zones, including from Himachal Pradesh to coastal Karnataka. I'm not going into detail, but you can see this the roadside, and then large ponds, and then salty land also. There's some wild rice grows. There are shallow ponds are there, and near the canals. Within the rice field also there will be few wild rice plants, and wild and BD are also there, which is called spontanea, which is a natural cross between the Uraja sativa and Uraja nivara or rupi program. Also in the urban areas, which has been used for the urban development and the industrial development, lot of wild rice is there, but it will disappear when the house is made or when the industry comes. So this is something resource we are losing it very very fast. In fact, this is a great germ plant resource which we are losing, and due to development. If you say in the single word, development is the enemy of these wild resources. And farmers also are not savior of that, because for farmers it is a weed. So farmers also don't want wild rice, and development people also want their enemy of wild rice. So it is squeezed from both the side, and only the geneticists and breeders, they can appreciate its value. So within time one must collect it and preserve it, because they have 10 times more variability than the land races. So we characterize this at morphological level. We have made a database of all these things, and we evaluated for drought tolerance. We found certain lines which are more tolerant than others. See, very highly sensitive to highly tolerant wild rice we have. We have done allele mining for some of the known genes, and we found better alleles by DREB1F gene. We found a better allele, and this paper have been published in paper. Published means not the paper is not the output, means paper has been peer reviewed, but that's a recognition of whatever work we have done. It has been reviewed by the people and it is recorded in a peer reviewed journal. So that means this is the work is that we identified a better allele of DREB1 gene, and the paper is the proof of that. It's not that we published a paper, that is not the thing. So we found this thing. Then anaerobic germination, like for direct seeded rice, is very important. So we found lines which have more than 90% germination, as compared to only 60% germination of the best check of the cultivated variety. Then we found lines which are better surviving ability under submergence tank, Here is one of these lines. Then two lines we found out of about 300 we have screened, which are better salt tolerance than the Pokhali. So in the wild rice resources, we found lines which got better trade value than the best of the land races are cultivated. Now, for every trait, for any trait we have screened, I shared it with other scientists. They have screened it for BLB. There are 13 lines which are all very highly resistant. We have to still do the gene discovery for that. Then for sheath blight also we found lines which are highly tolerant. And again, association mapping for salt tolerance was done, and we have found certain alleles which are better associated. Of course, this has to be followed up and taken up to variety level. One of these lines which I showed you, the salt tolerance line, MKSWR173, it was collected from the Himachal Pradesh, in the Mandi district in the Himachal Pradesh, because this was growing in they have some salty mountains actually. I was surprising that how come you have salt tolerance there in the Himachal. Then I was told that these mountains, they are salty. So this one we crossed it with IR64 and developed a mapping population to cut this is a standard method. You cross, you have the segregating population by parental, then you can, here we made one back cross. This is the standard method for advanced back cross method for mapping this. Whenever you have the genes are there present in the wild rice, wild relatives like in the tomato, it was done from pimpelifolium. And here in case of, we used the very similar approach here to BC1. So just like if you have the F2 derived F3 approach for mapping in the F1, F2, F3 mapping, similarly you can have BC1 and BC1, F2, you can do the phenotyping, and BC1 genotyping. And you can see this is segregating for different wild rice trait, like the color and the arms and the seed size. And also we phenotype for the salt tolerance, both at the seedling stage and also at the reproductive stage. Seedling stage was one a single measure injury score from 1 to 9 scale. You can see the distribution here. And then we use our rice genome chip to do the genotyping. And here, because it is the backcast population, we have to look for the markers which are segregating 1:1. So because we have 50,000 markers, then many markers will be not behaving themselves. So we take only those markers which are segregating 1:1 in the expected manner, and using that you create the map. So having large number of markers is the advantage, that out of that you can even have out of 50,000 even 1,000 markers are behaving in a map proper way. First thing, they have to be polymorphic and they have to segregate into 1:1 manner. So you don't have to have a separate step of polymorphism survey. In one go, you did polymorphism survey also. You found the markers and you made the map, and then you got the QTL here. So on chromosome 1 and chromosome 3 and chromosome 12, we found a very good marker. And some of the alleles are coming. The red ones here, you can see they are coming, they are contributed by the wild rice. The tolerance allele is cultivated by wild rice, and the green one, blue one, is contributed from the cultivated rice. So even the cultivated rice has got certain better alleles as compared to wild rice. So this is the situation, the QTL situation, where the trait is governed by multiple genes, and each gene has got the desirable allele and undesirable allele, and this can be distributed. That's why you get the transgressive segregation. So, and the job of the breeder is to bring all the good alleles into one variety. So here we have to just dissect it out, find these QTLs, and we have found it. This QTL on chromosome 3 is very good, and it's a new also. So we are just transferring this. We have made one more backcross, and we are trying to transfer it into cultivated variety, which will be in addition to the salt all QTL So in case of the salt tolerance, there only saltal QTL is known. There is no reproductive stage QTLs. Through these programs, we have identified one from the cultivated rice, and this is one from the wild rice. And now we are trying to mobilize them into variety. So I'm coming to very close to the end of my presentation, and I've tried to show you that how the basic research is very important, but its utility was only proven when the product is delivered. So these are through the QTL to variety project. There are four varieties of rice have already been released here by marker assisted by cross breeding. There is a Samba Masuri sub 1 with two genes of drought tolerance were added, 2.1 and 3.1, and this was released as a DRR Dhan 50 in 2018 by CBRC. Then Ranjit Sabban, Bahadur Sabban, released by Assam State, very highly popular variety, but they are flood sensitive. Sabban gene was added. Similarly, Swarna Sabban was already there, but two drought QTLs have been added. So now we have the varieties which are both drought and flood tolerant. And you can see here the picture, the farmers in Kushinagar were very happy that it yielded more than 5 tons with just one irrigation. 2 years back there was drought-like situation, and the original Samba Masuri was dried actually, but this variety yielded. So we are distributing the seeds to the farmers also, and almost 1 dozen varieties are still in the pipeline. Every year, one or two are getting released, either through state or through CVRC. So this is the kind of things we aim that finally, whatever research you do, it has to reach to the society. Then only it has the value. Otherwise, it will not have that much of value. And you can see the farmers. We are taking these seeds. We are distributing small mini kits to the farmers under the SCSP program. Government has created a program where you can spend money for extension activities, and we are making use of this. And this is very well received by the farmers. So in the second phase in the QTL to variety, we want to add this yield QTL plus the disease resistance QTL plus training part is there. And then this last slide I have here is that what are the future challenges and opportunities in this area, particularly the take home message which I want to convey is that the genome sequencing is very important because it has huge application both in molecular breeding and also as a even in the transgenic also, because when you design the probes for genome editing, there is no cross homology in the entire genome. How will you do it? Unless the genome sequence is available, how will you determine that there is no off target? So the having genome sequence is very important for any kind of the breeding, precision breeding application, be it the transgenic or be it is a molecular breeding application. So we need to decode the genomes of our crop plants, whether it is fruit or horticultural or field crops or pulses or legumes. If it is not there, then we need to do it. If it is draft, we need to reference quality. But having a good quality genome sequence as a reference is very important. You have seen the examples, how it is benefiting the rice community. Then, how will you do that? At the present state of affairs, we need to have a combination of long read, pack bio, or nanopore if it is coming, if it becomes useful. It's more refined, still it has got more error. And then we can combine with the optical fingerprinting or high C. There, you don't need to use in a single technology. You have to use multiple technology to make our assembly really acceptable and high quality. So we need to have for the fingerprinting, optical fingerprinting or high C sequencing and the long reads plus the short read also to finish it. Basically, we need a single molecule sequencing and single molecule DNA fingerprinting or optical fingerprinting. So these combination give you very high quality reference, finished reference genomes. Then we need to do RNA sequencing, full length RNA, once again single molecule sequencing, so that you can do good annotation of the genome, that each gene how many proteins it produces through alternate splicing, and that helps in the annotation process. Then we need to create high density genetic map. Of course, genetic maps help to anchor the physical map, because eventually we will have integrated genetic and physical map. So genetic and physical map become same. Physical map is also the genetic map, but in the beginning, you need genetic map to anchor this physical map. Then plant biology, we need to understand the plant biology at all levels. Its physiology, its biochemistry, its stress response. Those for the practical application, these traditional disciplines once again become important. The biotechnological tools you can use to understand the basic biology of particular trait which you want, a particular quality trait, stress resistance or disease resistance or yield per se. Then we need to characterize, our land races. We have germ plant, we have NBPGR, and we have large number of wild relatives which have not been collected. I think we should go in the field and collect even for the others. Like India is the center of origin for say pigeon pea and so many legumes. We need to collect those things before it gets lost. And then we need to do marker trait association studies, whether it is bioparental population or is it GWAS or Magic or whatever population we have, and NAM population. The purpose of all those populations is that to associate DNA with the trait so that it can be used in the breeding program. And so all these functional genomics tools we should use to identify the useful genes from our biodiversity to make use in the breeding program. And to do that, we need to have a broad knowledge of all the disciplines of agriculture, including its agronomy, whether it is agronomy, plant physiology, or soil science, biochemistry, or social science, everything you need. But this molecular biology or biotechnology or genomics is only a tools to aid those things. It is not the panacea of answer to all the problem, but it is just a tool. So with this, I'm not sure whether I took more times or less time, but thank you all for your attention and hope I was able to convey. If there is any question, I will be able to answer. I don't know how you are getting these questions. Um, so I end it there. Thank you so much.
Hello, Dr. Saab. Thank you very much for your nice presentation. Now, session is open for the discussion. There are some few questions from audience. I am reading it. First question is, can we get the PPT? Most of the audience are asking this question. And hello. Second question is, how sequencing is possible without PCR? Do you hear me, sir?
I think his mic is muted.
Hello. There are few questions from the audience. Hello.
Yes,
Do you
I
hear me?
can hear you. I can hear you. Can you hear me?
Hello.
My mic is not muted. My mic is open. Hello?
नहीं म्यूट नहीं है.
Can you hear me? मेरा mic mute नहीं है,
Hear
but
you.
I heard your question, but
Sir, I'm giving you in box, sir. Question box, sir.
can you hear me?
Yes, sir. And the question
Okay,
give you.
then I will answer. See, the question was that can you do sequencing without PCR? The first question was that can I share the PPT? Yes, you are free to share it with all the audiences. And
There are few questions.
the question is that how can we do sequencing without PCR? So
Yeah.
third generation sequencing, which is the single molecule sequencing, it does not require PCR amplification. Okay, it does not require PCR amplification because the single molecule you isolate the DNA and that DNA is sequenced directly. in the SMRT cell without PCR amplification. Okay. So in the first generation sequencing, you needed to clone the DNA in bacteria before sequencing. In the second generation sequencing, the cloning was replaced by the PCR amplification. And in the third generation, which is single molecule sequencing, there is no PCR amplification is not required. Just DNA is sequenced directly. Any other, sir? My question is: do transgenic crops pose major threat? Okay, this is a very good question, and this is the question many people ask in India and also around the world. See, transgenic crops, uh, they go through very detailed biosafety evaluation process. The whole question comes because we have the ability to transfer genes from across the species boundary. Like we take a bacterial gene and put into plant, for example, Bt gene. That's why this question comes into mind, that is it safe or not. So of course this biosafety evaluation is very important. It has to be done, and then after doing all these necessary exercises, then only it is released. So when any material which has gone through the regulatory process, one can be sure that it does not pose any threat or any danger. And whatever GM crops have been released for commercial cultivation, they are very popular, and there are no examples, to say that they have done harm as far as the threat is concerned. This is just one more method of doing plant breeding. And when it comes to genome editing, the most modern tools, they are even more precise. Like you can just make changes like the normal evolution process. You don't need a foreign gene. Even within the present gene, you can do minor editing to make it a better for you. Then either please tell what is the latest software or the preferred software used for physical mapping and QTL mapping. Well, there are many software. You cannot say what is the latest one. The QTL cartographer is still very popular software, but there are QTL networks are there. Many R packages are there. We conducted training actually. We conduct a NGS CAPT, ICR CAPT training every year. 25 scientists from our NARS systems to giving details in that. Um, Physical mapping, of course, uh, this software optical fingerprinting, then the software is already there within the part of the package. Otherwise, earlier FPC like fingerprinted contigs and LTC, this kind of software being used. How to categorize land races and wild relatives of crop species? You mean if I understand, you want to ask whether how to distinguish whether it is a land race or wild relatives? Well, that's a very good question, particularly um, in case of rice. The Oraja sativa, difference between Oraja sativa and Oraja nivara, Oraja rupibagan is very little. Most of the wild relatives, they have typical characteristics. Um, they will be having some kind of scattering in nature, or some physical property like long awns, are more bushy in nature. Sometimes they are easily crossable, sometimes they are not easily crossable. So one of the is there is a group of interbreeding individuals. If it is not crossable, definitely it is a different species. But in case of the rice, these are easily crossable. Similarly, in case of the pigeon pea, also you have Caginus caginifolius, like that. So you have to take help from the taxonomist. There is no single answer. So the relatives of the crop plants are known. What are the related species? So taking help from the taxonomist of that particular crop, you can uh, take the tips. In fact, when I started collecting the wild rice, I didn't know myself that how I'm going to distinguish. But there are certain key features were there. It has very long ounce, and it is uneven maturity, it is shattering, and I was able to collect lot of these uh, Varaja Nivara and Varaja Rupipogan, and there are certain intermediates are also there. If it is a crossable, easily crossable, then in nature itself the crossing has taken place and you get the what is called Varaja sativa spontanea. This is nothing but what is called the VD rice. It is an intermediate type. So that can happen in any crop, other crops also. So you have to consult this particular crop specialist for that. Then the question is that is SNP markers are equally important for biotic stress in plant? Yes. The answer is yes. SNPs are independent of the traits. They are just a marker. So it can be marker for the biotic stage, for abiotic stage, for quality, for yield, for anything. This is just a variation in the genome. Usually they are biallelic in nature, and you can use high throughput genotyping tools where you can do large number of genotyping, SNP genotyping. You can also do by sequencing, genotyping by sequencing for SNP. So you have to choose very careful which particular assay is desirable for your particular situation. Whether you have few SNPs, large number of samples, or large number of SNPs in few number of samples, that will determine which assay, which platform you have to use for SNP genotyping. What is the government plan finally accepting GM crops for utilization? See, the government plan is simple. Government is not against GM crops. Otherwise, we will not have the projects on GM crops. We have Department of Biotechnology. We have the regulatory authority is there in place. So government is not against it, but government wants to make sure that whatever product comes is the safe, and government also has to address the public concern that if the public is coming and opposing, then government has to harmonize all that thing. You cannot go forcefully. So it is being done actually. Even genome editing guidelines are being revised, and some of the genome editing product like SDN1 and SDN2, which involve very minimum changes, will hopefully be out of the regulation, as they are in many countries, including Australia, Japan, even Norway, which is very strict. They are many countries they say that is SDN1, SDN2, which involve very minor changes and which cannot be distinguished from the conventional mutation, they should be free from regulation, including even the BT brinjal. You actually don't know. Maybe it will come because in Bangladesh it is being cultivated. So these things they take time actually because of mostly social pressures. But as a scientist and as a government policy, it is not against GM crops. It is for GM crops with proper biosafety testing and evaluation and taking the community into confidence also. How heterozygosity create problem in assembly base, explain it. Okay, I mean, see how you do the assembly. You have the sequence reads, and when assembly, the sequence reads are maybe 100 base pair or 500 base pair or 1000 base pair. So the assembly software, what it does, it looks for the overlap. See, part of the sequence, it is overlapping, and then it joins them like that. So this overlap, it is 100% match. Mostly the most of the assembly software, they will go for the 100% match. If it is 100% matching, then you will join and create a context. But in the heterozygosity situation, the matches will not be 100% because the chromosome coming from the maternal side is different from the coming from the paternal side. So that's why it creates the problem. That's why people go for the inbred line and double haploid lines where matches will be 100%. So not much time to go into detail because of the assembly process. This is the problem. Unless you have the 100% match, you cannot assemble them. And to allow for the mismatches, different software has been created now. They can separate the maternal chromosome from the paternal chromosome, and then you can assemble them easily. Is molecular breeding is far better than transgenic crops? I don't think so. None of them are better or worse. Both are the tools and both are equally good. Transgenic is applied where the variation is not present within the germ plant, so you can introduce from the any other crops. Okay, and I think we can look for a time when these two will merge. Like you can do molecular breeding using transgenic. Suppose transgenic can be also used to create the isogenic lines, just like what we are doing by back crossing foreground background selection. If you know the gene, SD1 gene, you clone it and just you transfer in the best variety. That is, you create a nil by using transgenic. So none of the method itself is not good or bad. It has its own room. It has space for the application. So we don't think that way, that which is better, which is worse. Both are patent technique. You have to find which one can be used where. Okay, I think there are
Thank you
more
very much, sir.
presentations are there waiting.
There are. Thank you very much, sir. Now there is
Thank you so much.
other present of Dr. Sri Bhardwaj, Principal Scientist, Division of Genetics, ICR Indian Agriculture Institute, New Delhi. First, I am introducing him. Uh, he is a principal scientist and he is having a lot of experience in chickpea breeding. He has he has developed 8 chickpea varieties having climate resilience. He led the team of chickpea to release the first variety in pulses in India, USA 10216, developed through the marker assisted selections. He has led and also leading several multidisciplinary and multinational grant projects during last 20 years. He is the principal investigator for the prestigious US 8 million ICR Bill and Melinda Gates Foundation project on application of next generation breeding, genotyping, and digitalization approaches for improving the genetic gain in Indian step crops. He leads the several other projects in collaboration with the AgriSat, ACARDA, uh, UWA Perth, and University of Saskatoon, CGP, CIMITS, and the SORTS. He is also the AgriSat steering team member for GOBII programs. He has also published more than 120 research papers in peer-reviewed journals. He is core committee member of developing the descriptor for Biodiversity International BRL trials of DBT. It is so. I'm requesting Dr. Bharadwaj to present your uh,
Uh, Thank you, Dr. Khandelkar, for the nice introduction, and also I put my accolades to Dr. N.K. Singh Saab, who has actually given a good overview of how these technologies are actually helping, how the decoding of plant genomes is actually going to lead. Now, my lecture would be more of a practical approach, how these technologies, as he said, or as Dr. S.K. Rao, Honorable Vice Chancellor, has said, could be put down into the practical aspects, how the genomic technologies, and I'd be concentrating myself more towards the stress tolerance. I shall be sharing my screen, your entire screen, right? Share.
Yes, sir.
Hope now it is being seen. I can continue.
yes,
Yes,
sir.
sir.
Yes,
Sir.
Okay.
carry on.
Friends, I would be speaking on crop improvement, basically with respect to salt tolerance and how the genomic assisted breeding would be helpful. Whenever we speak of genomic assisted breeding, let me tell you that we have a large amount of techniques and technologies that can be used. And this particular lecture series which I'm presenting is a synergist of works from ICRISAT, from IRI, from CRRI, Katak, Meenakshi Sundaram, Dr. Gopal and Dr. A.K. Singh, Firoz and Hari Krishna, and many of my lab mates. It tries to put forth how this particular works were useful. Now, if you just look down, the slides are from Vashne, which says that we require 5 P's whenever you are looking to meeting the future generations' needs. And that is basically working towards the sustainable development. Whenever we speak of sustainable development, what we mean to say is that we are not compromising the needs of the present, but at the same time, we are not jeopardizing the needs of the future. Be it of the people to end their hunger and poverty, be it of the people to ensure their prosperity, fulfilling the lives with harmony in nature, be it of the people to work towards peace, be it among the nations and institutions to work towards the partnerships, or be it to the sustaining of planet. Friends, in this COVID era, we have seen that people did go out to buy cars, they did not go out to buy gold. But the first thing that they had gone out was to basically look at their food needs. So this is very highly inelastic in demand, and rightly said by someone that the future wars are going to be war of food or war for water. So these are the major challenges for international agriculture. Population is bursting. In every 3 minutes, we are adding about 12 people. In an hour, 1.2 billion people. And China and India are contributing in a very big way. How do we feed these millions of mouths? And at the same time, how do you provide employment to so many hands? The resources are getting scarce and scarce. If you look at the water resources, whether if you are looking at the decreasing limited arable land or degrading land in terms of salinity, in terms of increasing drought, in terms of increasing flood or the changing climate, and at the same time increasing poverty. So these are all the big challenges for international agriculture. And this big challenge is in way of providing more food with using less resources, maybe less arable land, less water and less labor. At the same time, we need to understand that every breeding process that goes into the development of these varieties is also not only time consuming, but also consumes a lot of what you say value in terms of money, value in terms of time, value in terms of decision making. And these three need to be solved if you want to have a greater deployment. And that's what the crux of today's lecture is. How translational genomics would be helping to have a sustainable agriculture. Well, we had, whenever you're looking at, when I say development of a variety or development of a gene pool, basically the fundamental region which we look down is creation of variable. Whether you are using a selection process which a particular breeder uses, it will be only effective if there is sufficient amount of variability available. In the previous times when the breeder used to do it, he used to depend on his visual selection. Slowly the biometrical techniques with Sir Ronald A. Fisher has started in 1940s to the what you call as later the cytogenetic techniques followed by the molecular techniques. The whole idea is, or the basic crux is to create this variability or know where this variability exists. And that is what you are going to do when you are trying to use the genetic resources. You see, therefore, you need to understand how much of this variability exists in these genetic resources. You are able to understand how or where it is tapped and how to use it. And for that, you may need to actually identify genes. That's how the famous adage has come, from genes to genes. I mean to say, from the genes that you wear to the genes actually are able to exploit. So at the level of making the course or at the reference sets or making the mini course or later where Dr. Gaur would be speaking on creation of nested association populations, or the multi-advanced generation intercrosses, or the magics, or development of rills or ills, which Dr. N.K. Singh has spoken. So you require to have a germplasm, you need to have genetic resources and to identify it. So almost the genomic assisted breeding is an amalgamation of different disciplines, whether it is from the gene bank person or utilization of land races and cultivars using the computational and functional phenotyping. Then these are now used to identify the genes which go down. But just identifying the genes would not help. You need to make crosses and finally develop the climate resilient varieties, either using the marker assisted recurrent selections or the marker assisted selection and the genomic selection. I would be basically concentrating here on marker assisted backcross breeding and the genomic selection that we are doing in case of chickpea. But you should understand that all these tools help you in identifying the genes, utilizing them in pre-breeding activities or all those breeding activities that make a line amiable to be used, and then this needs to be evaluated. And that's what I put a degree of caution, that you require a good amount of phenomics or phenotyping. If your phenotyping is not precise, what happens basically is you'll be developing what you call as a molecular phenotype. That means you'll be having a lot of QTLs for Delhi region. You may have the same number of QTLs for Hyderabad region, and finally the breeder will be confused to which QTLs you need to use. So the most important thing is you need to have a good phenotyping facility and at the same time validation of the identified genes, markers, and details. So we are coming to an era where we are coming to the 5Gs for crop genetic improvement, which is again from Varshney, where it has been already published in Current Opinions in Plant Biology, where the 5 genes refer to the genome, germplasm, genes, genomic breeding, and the latest coming subject on gene editing. While the first 4 fall under the gambit of genomic assisted breeding or using of the genomic tools, the gene editing is recently a novel one which is catching up with fans. Now, if you look down, for example, you need to have the genome assemblies for a given crop species, and that's what the whole talk of Dr. N.K. Singh was revolving around. If you don't have the basic information on genome assemblies, you will not know which of these genes or which of these characters which are linked to these genes and what is the methodology to be used. Now, it is as simple as speaking like this. The same group of subjects or your entries, when they are actually subjected to different statistical interventions, form different groups. So you should know basically for what you're going to use. Maybe let's take a class of about 100 people where there are male and female students. When you apply gender, maybe only 2 groups are formed. The moment you start applying, for example, height, 5 groups, same amount of subjects. The same when you apply from the states, they may come, maybe 7 groups. You see, the subjects haven't changed, only your intervention of the technique has changed. So you should know what technique you're basically gonna use them. So more than that, you should have the background information of this subject. Here, when you're looking at molecular techniques, you should literally have the information of the genome, and the genome discoveries have of late been erupting, and a large amount of data is being available in the public domains. But just having genome information would not stop. You require to have the germplasm characterized at the genome and the agronomic level, and a functional analysis be done so that you identify which are the genes and what are the characters that they're going to play, or you identify the QTLs, you have validated the QTLs in a different background population and have identified those which are tightly linked so that they can be used in the marker assisted breeding programs or marker assisted recurrent selection programs or any other type of programs. So a basic knowledge of these particular breeding approaches is also essential, particularly for the breeder who wants to jump into these programs. Whenever you're looking at the genome breeding approaches that are basically working, whether it is marker-assisted selection, whether it is marker-assisted back-cross selection, whether it is marker-assisted recurrent selection, a forward breeding where you are going to only keep in concentration the most important QTLs that have been identified and linked for a particular diseases or pests or for yield, which you would like your base population, which you are going to improve to carry upon, or the haplotype based breeding or the genomic selection. Now I'll give you an example from chickpea where we have been using, where in case of chickpea it has been reported that we have a large amount of lines, a good amount of phenotypic diversity. You have the Desi and the Kabuli types, but the genetic gains when we are using these lines is low. All the breeders are well experienced. They've been using the morphological means of understanding the diversity, maybe using the melanobasis technique or using the Euclidean principles. And though they were crossing, they couldn't get. Now, what happened is we thought we need to look exactly at the molecular level. What is the amount of diversity in the chickpea germplasm in the Indian subcontinent vis-a-vis what we get in the Wana region? That is the West Africa and the North Africa and the West Asian region. So what we have done is we have collected a group of lines which represent most of the accessions of the country as well as from the Wana region. And when we started looking at the molecular basis of this particular analysis, we could see that all those lines obtained from the ICRISAT gene bank or the varieties that were well preserved in the National Agricultural Research System actually branched totally different when you compared with the wild species or the land roses or a particular derivative. For example, PUSA 1103, which is the only line that has been released having a wild species in its accession, as well as those of the Icarda origin. This clearly indicated that greater genetic gains can be obtained by crossing these lines from the Wana region with those of the Indian subcontinent. And we started looking at these land races. Someone was asking in the previous lecture, how do we distinguish the land races from those of the wild species? To add upon to the thing that Dr. N.K. Singh has put forth, the wild species belong to primary gene pool, no doubt, but mostly they belong to primary gene pool B or primary gene pool A in case of where they're single way crossable. For example, you can easily cross reticulatum with eritinum, that is cultivated chickpea, when you use cultivated chickpea as the female parent. In case of land races, you can cross either way. So that is one more easy way. Apart from, of course, the wild species have an wild type of character. Most of the land races are following the same character as the cultivated types, but these are known since times immemorial, or they have a long history before the breeding program has commenced, which we actually put down. Now, if you look down, we have collected this amount of, in our Icarda breeding program, a large amount of land races of chickpea from all over the world. And we basically were looking at their process of how they were actually working towards salinity tolerance. And we did these studies both at IRI and CSSRI Karnal along with my student Snehapriya. Earlier, the genetic studies were done by Neeraj. We have developed a rapid phenotyping technique. As I say, a good phenotyping technique is prerequisite, and at the same time, a reliable phenotyping technique, which you can do at your place, is a prerequisite for all genomic activities, and we could identify clearly donors. And when we started grouping of these land races, you can very clearly see that the universally accepted ICCB-10 by the already published sources groups down with these land races, the Icarda land race chickpea line, different line numbers, 0 to Czechoslovakian or to those from some of the Bangladesh or the Greek continents. Now we started looking at actually how these things worked on. And we started trying to mine out some of the alleles which were responsible for various processes, either they are for relative water content or membrane stability index, which Dr. Neeraj has done for his graduation work. And we have seen that basically 3 type of genes are upregulated, and we have seen the MIP transcription factor and dehydrin, which actually plays a major role in case of membrane stability index, was upregulated in some of the cultivars, whereas the CAD gene was upregulated in some of the cultivars. That means no single gene or no single pathway is responsible for all of them, and you need to pyramid these activities. And at the same time, like Shiv Agrawal has indicated, that while the process of domestication occurred from the wild resources to the land races, or the process of breeding has occurred from domestication to that of modern cultivars, what we see is a large amount of non-selective neutral genes. Maybe these are the buffering genes, maybe these are the adaptive capacity genes were retained. And at the same time, selective genes of domestication basically decreased. But the most important thing that you need to know, that the genes for crop improvement drastically reduced. Actually, so as a result, what happened? The diversity in the modern cultivars has drastically reduced. And this throws another light that you need to actually move towards looking at either the wild species or towards the land races and actually tap these genes. See, that's what happened in case of tomato breeding. If you understand, the QTLs for seed size, when you actually compare the QTLs for the what you call as the tomato size or the thickness, have come from wild species like Lycopersicum peruvianum, which are very small fruits, but the seed thickness or the flesh thickness, the QTL were basically obtained and transferred. Now, when we were actually fingerprinting these land races using these genes, we basically could identify, which was done by, again, some students, one of my students like Shubha and Rinki Madan, that we could identify a set of genes, for example, the MIP transcription or even Ascochyta blight QTL1 as well as QTL3, and similarly the dehydrogen gene analogs. So this is where we started looking, can we start using them in forward breeding. There are about 3 approaches which are widely used, whether it is marker-assisted backcrossing method or marker-assisted recurrent selection. Of late, the genomic selections. One easier thing is they don't have the issue of regulatory approaches, and therefore it can be easily practiced and they have a wider acceptability. When I'm saying it is easily practiced, we have moved a long way. When you look down, we have moved a long way from that of RAPDs or even isogynes when I was a student at this own division working, to thus the present state of SNPs, to that of almost sequencing of the plants. So you can do the same amount at a lower cost, right? Therefore, your ability to screen the initial segregating populations, even at a nascent stage, that's well put. Like, what does plant height do? Right, but then you can actually identify for lower plant life at seedling stage and call out others, like Dr. N.K. Singh has put forward. Now we'll be speaking of some of the case studies that we have seen. For example, in case of bacterial blight, this is from Dr. Meenakshi Sundaram, which is a major production constraint in rice. It's a major production constraint caused by Xanthomonas oryzae, Petavar oryzae. It leads to a huge amount of damage, 10 to 30% damage, particularly in the irrigated and lowland ecologies. And basically, we have been using resistance breeding. But then when you look down into resistant breedings, there are about 40 resistant genes, and the peculiarity or Vidambana is a single R gene containing varieties, it becomes susceptible. There are large type of pathotypes, more than 22. And almost, if you look, all the rice growing areas, the major rice growing areas are totally affected by this particular devastating disease. And then the work, both in case of non-centered rice at NRRI, Cuttack and other groups, And for the center, the basmati rice at IRI, led by Dr. A.K. Singh and his group, have done beautiful work, and perhaps were the first to release the marker-assisted backcross-bred varieties having bacterial leaf blight as well as other resistances. Now, what basically Sundaram and his group has done, they have taken the Samba Mashuri, one of the most popular variety, BPT 5204, popularly grown in Andhra Pradesh, most preferred by farmers because it has excellent grain cooking, and more important is it has market preference. It has a high market preference. It has a large area, more than 3 million hectares is under cultivation, but it is highly susceptible to bacterial blight. And therefore, a need was necessitated in this particular that we need to develop bacterial blight resistance. Then they did have a particular donor, WS 1113, which has both the genes, three genes which are responsible for BLB resistance, XA21, 13, and 5, which were actually obtained from the background of PR106. They have been developed, which has been done by PAU. And it is resistant to bacterial bite. But when you look into the grain characteristics of chalkiness or the plumpness, it is not preferred by the non-centered growing, what you call as areas. So it was serving as one of the donors, and they followed the marker-assisted backcross selection, where for the foreground selection, they've used the link markers, and for the background selection, they have used those markers which were tightly linked to the Samba Mashuri. And then finally, what they could do is they could develop an improved Samba Mashuri. If you look down at the leaf scores, the improved Samba Mashuri has almost a less streak of BLB infection, whereas when you're looking at Samba Mashuri, more than 50%, that means it comes under the susceptible characteristics where bacterial blight, it is highly susceptible. Not only that, it is a fine grain type with excellent cooking quality, which has high yield of more than 5 tons, and most important is it has a low glycemic index. And this has been already receiving large amount of accolades. Mind you, no work a single institution, and this is one of the best examples of multi-institution collaboration between the CSIR laboratories, between the ICR laboratories, and giving down into a final product that is already being licensed, and not only earning money to this particular group of institutions, but also providing, most importantly, biotic stress resilience in otherwise the bacterial leaf blight hotspots of the country. Now they are looking at various other genes that can be introgressed. For example, be the blast gene, where they were looking at Pi2 and Pi54, or brown plant hopper, another big nuisance, BPH33, or the Galmage genes GM4 and GM8, or looking at the abiotic stresses, for example, submergence tolerance sub1, looking at the drought tolerant or the salinity, or looking at how to improve the nutrient efficiency in already low soil phosphorus conditions of the country in the rice tracts using the PUP1 genes, or in general enhancing the yield using the GNA1. Now similarly at IRI, they started looking at combining the basmati quality characters by Dr. A.K. Singh and his group with the bacterial blight resistance, and a large amount of derivatives as well as released cultivars have been done. This is the first work anywhere in the country. This was done with along with a close collaboration between IRI and National Institute of Plant Biotechnology. And we could see improved Pusa Basmati 1, for example, which is having the Pusa Basmati 1, which was improved, having the blast resistance genes 13 and 21, or Pusa Basmati 1718, which is again an improved version of Pusa Basmati 1121, which has the bacterial blight genes 13 and 21, or Pusa Basmati 1728. Apart from what you call as blight, they have also improved blast resistance. Again, two recently released cultivars where they used PI2, 54, and P19, which were basically the blast genes which have been pyramided. Similarly, they have also started looking at non-basmati rice varieties, where they started improving again for bacterial blight using the 13 and 21 pyramiding these two genes, or the blast trying to pyramid more than two genes. So these are some of the new cultivars that have been recently released. At the same time, colleagues Dr. Hari Krishna and others from the wheat group are looking at QTLs of grain weight per spike under drought and heat conditions. See, it's very good for biotechnological publications or initially discerning how the traits work when you are looking at drought per se or heat per se. But as a breeder myself, what I'm basically interested is how, because these two cannot be just separated individually, but they work together at the terminal stages in case of the rabi crops, whether it is chickpea, whether it is wheat. And we need to understand what phenomenon or what QTLs basically work, imparting resistance for drought susceptibility index per se, or these particular two traits. And they could actually look at the biomass index or NDVI or drought susceptibility index, which takes yield also into consideration, and could actually identify QTLs which are having a positive effect or the QTLs which were also having the negative effect, and look down. And then another particular stress that they were working on, again by Hari Krishna group et al., is how do you map an yellow rust resistance gene for the new races. In races keep on emerging, there is a boom and burst cycle between the rust resistant genes, which has been a menace, and it does not help the breeders as well as the molecular biologists and the biotechnologists to sleep over. Whenever there is a burst of these and we could get resistant cultivars, the viral strain itself mutates, and we have a new race 110S119, and the group could clearly identify two particular QTLs which were actually used, and they were trying to actually what you say is move this into a cultivated line. And then if you look down, because there are a large number of races that particularly look, and you can't actually miss down the basic characteristics, for example, reduced height. So they have used all these 13 positive alleles which were linked to this positive drought stress in the initial stages, as well as the stripe rust resistance, and also the dwarfness. And this is one particular line, HD3296, which could have these particular lines. Similarly, Rakesh would be speaking on the pearl millet genome sequencing program that actually is working down. And friends, we should appreciate that this is the first program in the country or world where it has released a particular variety or hybrid which was due to marker-assisted backcross program. Now, when you are looking at, as I say, unless you have the genome sequence, you cannot identify which genes are particularly linked. And once the genome sequence is available, the Dr. Rakesh group as well as Madam Tara's, you could see that a large number of QTLs have been identified. Let's say for high iron and zinc content, either using biparental population or for various agronomic traits, or using association panel for drought tolerance. And it's really appreciable that among all the coordinated breeding programs of the country, a baseline for iron and zinc has been introduced and well appreciated even by the international breeding programs, that all the varieties that have been coming out, or all the hybrids that are coming out in case of pearl millet, AACRP program have a minimum baseline, and only if a variety which is qualifying not only in yield but also for these two traits comes out. And it is not just by estimation. Now the breeders are started using how do we actually fortify, and now they are looking at bioavailability. So the second thing in case of cross-pollinated crop is, unless, as I said, you have a heterotic gene pools. Earlier, when we were using morphological techniques and diversifying or identification of diversity in these morphological gene pools, many a time the required amount of dominant heterosis was not seen, because many a time the morphological variability masks the molecular variability. This happens, as you all as breeders or students of agricultural sciences understand, that there is a G into E component, and E component plays a major role, and you need to do multi-location, multi-year environment testing to actually reduce this error variance. But this actually is decreasing the burden when you're putting the molecular tools. And as I said, perhaps this is one of the oldest hybrid which has been released in the national system using the ICRISAT-NARS partnership HHB 67. Now, similarly, a very good amount of work is being done by Dr. Firoz and his group at IRI, where they were actually looking at improving the quality protein content. Whenever we are looking not only at developing high protein maize, but they're trying to increase the provitamin content. Now, if you look down, they have actually used the popular varieties Vivek, and they have developed a series of lines for different agroclimatological regions. For example, QPM9, Pusa Vivek. QPM9 is the country's first provitamin rich maize hybrid. It has a large amount of provitamin A. Even the tryptophan and the lysine contents are very, very good. But at the same time, their average potential grain yield is equivalent to any of the commercial hybrids. Similarly, they have released for Northern Zone, which is again another major maize cultivation tract, Western Plain Zone. And then here, if you look, this is again a QPM hybrid, which has a large amount of tryptophan, 0.91, as well as lysine, which is 3.62. Similarly, HM18, again for the peninsular region, and we understand that most of this feed goes to poultry, particularly in the country. We all are aware of it. That means indirectly, you are increasing the feed value. By increasing the feed value, you are increasing the quality protein that the animal meat eaters get it through having the poultry that is fed with this QPM. This is what actually, what you say, US does when it imports our DOC of soybean, where we see that our protein content in the soybean decorticated oil cake is very, very high. And as a result, they use it for feeding for their bovine or poultry feed, and then they export it as best fed protein. The same thing, that means we are indirectly increasing the value of another product by increasing the value of the primary product that is going for its feed and also in food. This is one more that we have used, for example, Pusa improved HM9. Now, when you are looking at chickpea, where we have been using and developing molecular breeding techniques for improving drought tolerance in chickpea, an international consortia led by Rajiv and his group could identify the QTLs, which they could identify a QTL hotspot. Whenever I speak of a hotspot, that means apart from carrying the QTL for that particular trait, which was very much narrowed down, flanked by markers, this particular QTL hotspot also carries for seed size, also carries for harvest index, apart from carrying QTLs for root length and root volume or root density. Now, a large number of groups, including my lab at IRI, Paul Kimamoto's in Ergton University, groups at Australia, as well as other labs, were trying to introgress this particular QTL. For example, we tried it into Pusa 372. We could clearly identify two markers which actually sit within this QTL, NCPGR 21, NCPGR 127, again developed under a collaborative program of Indo-Australian as well as other DBT programs, which we are used for foreground selection, whereas we use the parental based markers for the background selection. And we have used our offline facilities at Dharwad. I thank Dr. Patel, who was actively participating even in genotyping and other activities. And then in a short period, normally this breeding program, because we take almost 6 to 8 years, we could achieve this in 4 years. And what we have basically done was, when you look at this particular thing, this is ICC 4958, which has a large root volume. You can see along with a long depth, and this is Pusa 362. No doubt, friends, you should understand Pusa 362 is not a poor cultivar. We did not use a poor cultivar. Pusa 362 itself ranked among the top 3 lines in an India-US program, where it was tested in 20 locations in India and 15 locations in Australia as one of the drought tolerant already line. And this has been, or similarly in case of Pusa 372, one of the most high yielding lines of central zone, has been a check for a long time. And if you look down, my colleague Dr. Sailesh, or when we have used Pusa 372 myself, we could develop lines. This was the BC2F1 pick that he has given me, which I'm showing, having the characteristics of already ICC 4958, along with, if you look down at the branching characteristics of our Pusa 362. And in case of Pusa 372, if you look down, when you look, we try to bring down introgress this particular QTL, removing the background noise from ICC 4958, preserving the basic characteristics of the Pusa 362. This is how actually when you're, and mind you, these lines which we developed not only competed with the recurrent parent, but in a trial of 10 locations, when you are looking whether we have used DCP 92-3 or JG16, GG4, JG11, GG1, these are the checks of various regions, and all these lines vis-a-vis checks have given more than 10 to 20 to 30% yield. And not only that, Dr. Mahendra Tudi has also developed a SNP marker, which was actually tightly linked to wilt tolerance, multiple zones, that is particularly to the south and central zone. And you see that we have a wilt tolerance marker. That means the basic requirement of having wilt tolerance for a variety or a line to be notified or to be identified in case of AACRP was ensured, and at the same time the QTLs for the trait were introgressed and the yield gains could be achieved. And this is the AACRP data that I am providing to you. And please look down at the moisture percentage at the flowering or at maturity stage of the test location in case of the most drought tolerant locations, which the PCASRP chickpea has identified. And when you look down, you see that BGM 10216, which we released as Pusa chickpea 10216, had almost a 16% average increase across the locations and under highly drought stressed conditions. And therefore this was the most drought tolerant variety that has been identified, and now it has been notified by the CVRC, where the QTL hotspot was introgressed from ICC 4958. The new variety not only gave, this is under the drought stress conditions, but under drought stress conditions it also showed equivalent to some of the commercial lines that have been released, right? And it has a very short maturity, 106 to 110 days, perfectly fitting into the cropping systems of the central zone for where it was released. It was showing resistance to fusarium wilt, dry root rot, as well as stunt diseases. And therefore, we could release this Pusa 10216 in a short time. Very good collaborations with ICRISAT from Dr. P.K. Jain, National Institute of Plant Biotechnology, my friends from IIPR Kanpur, Dr. Soren, Dr. Aditya, and we could culminate in releasing, and there are a large number of lines already in pipeline where we have greater amounts of yields, some of them being now entered into not only the special trials, but regular trials also. Similarly, in 2019, one more variety using the same QTL was developed by Paul Kimomoto and the group. In case of Ethiopia, the Gilatu is one of this variety. It has also 3,822 kgs, 3.8 tons at Nairobi, at RC and other locations, and it has given a 15% advantage check. See, now you can look 16% in my case over 10 locations or 15%. That means this is a robust hotspot. And such robust hotspots which have been validated need to be used by the breeders. The other line of work that we were working on was in a generation challenge program which was funded by CIMET and which was actually looked upon by ICRISAT, IRI, Indian Institute of Pulses Research, and National Institute of Plant Biotechnology in a NPTC program, where we were looking at a hotspot which carried almost 1453. These are the four QTLs for various races, and also Ascochyta blight, which was published by Teresa Milan. And we started looking, I'll move a bit fast. We started looking at introgression of this particular QTL. We could achieve it, and you could see this is PUSA 372. Again, we were introgressing into the same line because our idea, my colleagues, Chauhan or Rajesh or others, we were of the idea that we would like to bring down into the same where we have developed a drought tolerance, and our idea was to merge these. And in the foreground, if you see the tolerant lines, you could see the plots of these lines where you have the susceptible parent and the resistant lines where my research fellows were taking down the observations. So we have already entered. These particular lines are already in the second year of testing in the AVT2 stage, expecting some of them to be released. Already one has been released last year. And when we look down at these lines, when you are looking at the marker-assisted breeding program, either in PUSA 391 or our BGM lines, which we have, you look at the amount of yield under wild stress conditions. A huge amount of yield, then already the previously released Super Onigiri. Therefore, if we have actually validated 5 markers for foreground selection, which are tightly linked, and a new SNP marker from Mahendra Tudi. We have actually used a large amount of markers for our background selection and a large amount of lines in different backgrounds. We are not looking at one, but we are now targeting most of the lines which are called as mega varieties, having more than 200 quintals of breeder seed production. In case of legumes, it's a big thing. We could have 6 and 2 are entered. And this was last year that has been released, Super Onigiri, from the Manur by Dr. Manur and group, where they have actually brought out the QTL, which was responsible for the South Indian condition, having a 7% yield advantage. Now, similarly, in case of groundnut, Manish Pandey and group, because I'm covering almost all the crops, I was putting here that Manish Pandey and his group were looking at increasing the high oleic content in case of groundnut. And through the marker-assisted backcross breeding approach, they could actually develop lines which were having rust tolerance as well as leaf spot resistant. These were basically entered into the national trial of groundnut breeding program by National Research Center on Groundnut, now Indian Institute of Groundnut Research. And then they could actually develop the first high oleic acid specialty groundnut with ICRISAT and the NARS group, and they have been released. For example, the Girnar 4 and the Girnar 5 for the 6 major growing what you call as states of the country. So this is another, means in rice or in case of chickpea or in case of groundnut, we have excellent examples where these QTLs or various genes that have been identified have been deployed into the modern breeding programs. They have not been only just deployed, but the outcome is coming down now. But I certainly remember that it was the efforts of NTPC or DBT, DST or the ICRISAT programs which commenced way back in 2005 whose fruits are reaching. So certainly the basic studies, the genomic studies are very much essentially required. Same was in case of Pigeon P, where Dr. Raje's group, along with Dr. NP Singh, is looking into ways to alter the plant types using the genomic information that has been developed in case of Pigeon P. Now, slowly from marker-assisted breeding, we are moving towards a new breeding cycle. There is nothing called new breeding cycle. It's a breeding cycle, and I put it as a improved breeding cycle. See, whenever we are looking at a breeding cycle, what the breeder is interested is to increase the genetic gain of the base population. Unless you increase the genetic gain of the base population, it will be very difficult to actually realize the yield gap, reduce the yield gap one, and increase the genetic potential of your base population. When you are looking normally into the breeding program, it depends on the selection intensity. It depends on, it is generally 1.06 to 2.06. Selection accuracy with which you are able to select what is the amount of genetic variability or the variance that is present and how many cycles you can do. And now, because you can even increase the selection intensity, because you can chip in the molecular data or the molecular genotyping data, where from almost 80 rupees for one marker, we have come down to a panel which hardly costs you 20 rupees. Now you needn't have a very sophisticated lab. What all you can do, you have kits with which you can just extract the leaf and send to the service centers, either at National Institute of Plant Biotechnology or at National ICRISAT, the Center for Excellence in Genomics. You specify what are your basic primers or the QTLs that are linked, and they will do the selection and bring it. And within a 20 or 30,000 rupees, you can carry out the best forward breeding techniques. Now, similarly, now because you needn't wait to do it only in target environment. That's what we did in case of our drought breeding program. We did some of these selections even in off-season areas by Dr. Patel at Dharwad. You can greatly increase this selection accuracy and at the same time increase the favorable rare alleles or the alleles of this choice. And therefore, you can greatly increase what you call as the genetic gain or the breeding value, which I call as the new breeding cycle. Thanks for Gopal for giving me the slide where he sees that now there is a paradigm shift, and it is very important that we ourselves modify. I do agree that when there was a visual selection and people started moving to the biometrical selection, there were lot resistance and large amount of hiccups which were slowly overcome. And where people moved from biometrical procedures, they started moving with various progeny testing procedures. Again, there were a lot of school of thoughts. It's always good to have this school of thoughts, but at the same time, it is very good that you embrace the technology that has been validated so that you can provide value for money. Government or the particular organization actually spends a lot of money in developing one line which needs to have durability and sustainability. And this is how the particular breeding cycle or the paradigm shift in breeding is occurring. If you are looking down from almost 11 years of breeding of cycle to whenever you are using the basic marker assisted breeding to that of the genomic selection. Now, the difference between the marker assisted breeding and genomic selection, you need to understand that we are selecting parents which have greater genetic gain for your first round and second round of crossing. Therefore, you have a greater emphasis of all the alleles which are present in the elite group as well as your rare alleles that you would like to combine. Now, when you are looking at rapid generation turnover, which Dr. Gaur would be presenting, where they can able to take almost 6 generations, which has been published by them in normal greenhouses, to that of speed breeding by Lee Hickey in the crops, you can understand that when this particular genomic selection or marker assisted forward breeding is combined with this rapid generation advancement, the gains that you achieve to a breeding program are going to greatly increase. Certainly, you need, when you want to capture all these available common good alleles in a particular breeding program, in case of genomic selection, you need to have a genomic population or a training population, which is well again phenotyped for these basic traits. That's what we have done in case of our breeding program when we were working for our genomic selection along with ICRISAT. We developed a 320 breeding program, and these were actually the released lines or improved cultivars, as well as the basic fixed lines from the ICBT trials that we have taken from ICRISAT. And then we have genotyped over 2 locations as well as 3 seasons in 2 different environments. A good amount of work again done by my doctoral fellow, Dr. Rajesh. And then we have done a huge amount of genotyping in this particular thing. Earlier we sat with about dart markers. Now we have moved to SNP markers, and we could see, like, unlike other crops, we started making the prediction accuracy using either different models, maybe the Bayesians or the random forest, which gave the best prediction. And the prediction accuracy ranged from 0.89 to 0.91. There's a very high prediction accuracy. Now you should understand that for most of we are having again traits like drought susceptibility index, which is again what you call as a dependent trait. And generally we have 0.5, and if I as a breeder in my normal breeding programs have a selection efficiency of 0.6, I'm really happy. And if I'm really able to get this selection efficiency, it's a gold mine. But mind you, I am a breeder with second thoughts. So I said I should not stop here and blindly use it, but I should revalidate whether really this breeding prediction accuracy that I got through the random forest model or through the other models really works in the field, and let me compare it with my own selection accuracies. So basically what I have done is initially I started looking down why there were subtle differences. Then we saw that because we have used the Desi lines, Kabuli lines, and we have combined them to predict the models, there could be a little bit variations. So what we started is we started separating them and looking at their predictions, and we saw that Kabuli could better predict Desi for characters like seed size, whereas Desi could better predict Kabuli for drought susceptibility index, number of seeds. So we said let's stick on to basically the only Desi lines, where because 80% of these are the major chickpea growing regions. So we started looking at genomic predictions. But what I have done is I have, based on these genomic predictions, I have selected about 400 lines from the genomic population. And at the same time, using the visual selections, the plant architecture, the single plant yields, as well as the branching patterns and the post-harvest observations, like the seed size, seed coat color, I made about 200 selections, and we independently evaluated them in an augmented block design. And what I see that the prediction accuracies as depicted by the genomic predictions, the phenotypic predictions almost follow, and many a case you see that the predictions based on the genomic selection, they oversee or overcome the phenotypic prediction. What I'm saying here is that means whenever you are looking at the prediction accuracies using the genomic predictions, whether it is 91, we had a variation because we had rains in that season. Therefore, the flowering was prolonged in case of morphological or exact taking of data, because most of the flowering certain times get shed, and chickpea produces vegetable growth again once rains commence. So, or for the days to maturity, or for standard seed weight or seed yield, I'm sorry, for the plots. And you can look that when you are looking at lines which are very, very high yielding, the prediction accuracy was much more, 17 times or 200 times much higher when compared to the phenotypic prediction. I know this is a preliminary data, but if it is not 200 times, even if it is 10 times, that is a great gain to the genomic breeding program. So what I try to conclude finally is that next generation genomic approaches are very, very useful to develop climate change ready crops. For a given crop, there is a need to develop genomic resources. Totally agree with you, Dr. N.K. Singh, sir. We need to develop the basic information, that is genetic maps. We need to understand what are the pipelines, what Abhishek Rathore is doing. We need to also have genome sequence assemblies. Most important, the primary set of people, the gene bank curators also play a very major role, because you require to have germplasm collections, the breeding populations, the physiologists play a major role, because you require to phenotype them precisely for the interested trait with high throughput phenotyping and genotyping facilities. Again, what Dr. N.K. Singh clearly says, that you need to identify these particular gene clusters or QTLs that are very tightly linked, either using the GWAS approaches or the linkage mapping approaches or BCS-seq or QTL-seq approaches. Once you have well validated these, then you start using them in your genome breeding approaches, whether you are using the marker-assisted selection, the back-crossing programs, or the recurrent selection, or haploid breeding. And of late, it is well imperative that you improve your genomic selection approaches to improve your base population value. And that's what I put. It is not a work of my own, as I said. It is a synergy of all the workers across the country, across the institutes. This I presented, whether it is Dr. Meenakshi Sundaram from Cuttack, Gopal, Dr. A.K. Singh, IRI, Dr. Hari Krishna from Wheat, Firoz Hussain from Maize Group, Tara and Rajesh from the Pearl Millet Group, Manish Urkiwal from Ikrisat along with Raji, Manish Pandey, B.S. Patel, my colleagues, my students. I'm thankful for my work, which would not have been possible but for these funding opportunities. Thank you, everybody.
Thank you, guys, Dr. Sir. You have presented nicely and in easy language. So session is open for the discussion. There are very few questions. I'm selecting. So question will appear on the screen.
Speed breeding benefits, in which crops it is done in India, what are the main benefits, in which area India we can do this, how it is done. The speed breeding, whenever we speak of speed breeding, the whole idea is rapid generation advancement, right? So that you can take more number of generations in an area. Private companies like Corteva in India are routinely taking up for their wheat, maize, and other breeding programs. We have a rapid generation advancement in case of wheat as well as in case of chickpea or other crops, where of late we are using the off-season facilities. But now ICRISAT, because you require certain standardization of protocols, ICRISAT is developing a large speed breeding facility, a national speed breeding facility in its campus and even in the NARS system. We can apply it to any crop there where we can actually, by manipulating the temperature or by manipulating the light, basically, we can take more number of generations. Yes. Yeah, beautiful question. What is training population? You need to train your models, isn't it? That means you need to see the same set of population may not work for all traits. So if you are looking at drought traits, you need to have a population which is having these donors and the baselines, and where you can get a common set of genes, है ना, and which will predict. So that basic population which is used to know what is the prediction efficiency is called as the training population. Some people also call it as genomic population.
Uh, Now we are running late, so I am calling next speaker, Dr. S.V. Charu Ramamitra.
Thank you. Thank you.
So before calling you, I am introducing uh, your biodata here. You, you are working with uh, uh, you are a learned teacher dealing with the molecular markers, molecular breeding, advanced in molecular breeding and genomics. You have published more than 38 papers in different reviewed journals like Science, DNA Research, Plant Biotechnology journals, Molecular Breeding, etc. Your major contributions uh, contributed in hit jute and cluster bin whole genome sequencing projects. You have also developed a large number of molecular markers, especially SNP and SSF markers in rice, pigeon pea, cluster bean, maize, and rose. For the first time, use markers showing the segregation distortion in plant for QTL mapping for grain size traits in rice. You have a contribution in QTL mapping, novel QTL identification, marker designing, and integration studies. You have also contributed to the national initiative on the establishment of the National Repository of EMS mutant in rice as a co-coordinator since 2012. You have also developed and maintaining the database EMS grade N22 with the trade-based research features and genic sequences information N22 and basic morphological and SSR profile of the mutants. Now, I am requesting you to deliver your talk before the delegates.
Hello, am I audible? Am I audible?
Yeah, you are.
Okay.
Yeah.
Thank you, sir. Thank you for the introduction. And at the outset, I first thank the organizers, Vice Chancellor, sir, and the plant breeding head, Dr. Kandilkar, and also my dear friend as well as younger colleague, Sushma, who kind of always ropes me in whenever she does something. So I thank you one and all. And I also thank my co-speakers, especially Dr. N.K. Singh, sir, for delivering an excellent overview of the genomics, and then followed by Bharatwaj sir for the latest applications how for the plant breeding students. Now you have sir. Now with all your permission, I would like to most of direct my talk to students, student community, because all of us here, my co-speakers are very learned people. So rather than talking to them, I would talk to the real audience of this program. And sir, I am sharing my screen.
Okay, please share me.
Yeah. Just a minute. Sorry. Can
share
you see my screen? Sir, can you see my screen?
नो मैम, अभी आप शेयर स्क्रीन करके और जो स्क्रीन आपके कंप्यूटर में दिखेगा उस पे क्लिक करके शेयर बटन पे क्लिक कर दीजिए मैम.
Just a minute, sir.
Okay.
Shares. वो शेयर में नहीं है, that is not getting highlighted.
मैम शेयर पे तब आएगा जब वो जो स्क्रीन पे आपको दिख रहा है एक जो भी उस पे माउस पॉइंटर जाके क्लिक कर दीजिए तो शेयर बटन आपको एक्टिवेट हो जाएगा. ओके.
या या
ओके मैम.
Is it seen now?
एक मिनट में
You have to share
सबमिट कर देता
twice.
हूँ।
एक बार करने के बाद फिर दोबारा उसमें क्लिक
एक
करेंगे तो फिर शेयर बोलेगा, 2 बार करना पड़ता है।
एक मिनट। Yes.
Yes.
It is visible now.
Okay, ma'am.
It is visible.
It is visible?
Yes,
Yeah.
ma'am.
Now it is
Okay.
loading.
Then now
Yeah.
you have to present. Oh shit.
Oh, got disconnected?
नो मैम डिस्कनेक्टेड नहीं है अपना वो प्रेजेंटेशन फाइल पे क्लिक कीजिए मैम टास्क बार पे। यस मैम इसको फुल स्क्रीन पे कर दीजिए मैम वो प्रेजेंटेशन बटन पे क्लिक करके स्लाइड शो पे स्लाइड शो।
Yeah, it's happening.
Yeah.
Yeah, it has now come full screen. So far, from the morning, you have been listening a lot of things. One of the things that I would like to remind you, one of the things Dr. N.K. Singh sir said, what he said was that whatever we are doing, it is only the continuation of what we have been doing in probably with better approaches or with better new tools and techniques. So one of the new tools is in that way molecular markers, which of course not a very new tool. It is a 40-year-old tool now, 4 decades, because 1980 we started this. But this tool has enabled many things, and one of the things that is identification of genes and markers. And in that, if you look at molecular markers, we started with RFLPs in 1980s, and today, if you look at the talk of Dr. N.K. Singh and Dr. Bharadwaj, you heard of all the time SNPs. So it is the new rage, and people are almost everywhere, other than a few cases where we follow sometimes SSR markers also, especially in something like fingerprinting cases. Other than that, most of the work is being done with SNP markers only. So but when you look at in the natural population, when you go, whether it is a crop or it is an animal species or human species, any species you take and you measure any trait, you will see a continuous distribution rather than a discrete distribution. So this is the crux of all. So most of this means most of the traits are controlled by a large number of genes. That is why you happen to see a continuous variation rather than a discrete variation. It could be even otherwise a character like a color, which you can say presence or absence. You can measure it using a colorimetry, and you can record the color intensities. That will tell you it is not exactly a discrete trait. It could be also be treated as a quantitative trait. So from your when we take any natural population, we can see this, except for some cases like blood group where you have discrete classes. Otherwise you will always see a continuous distribution. So from your courses, you must have learned that quantitative traits have like they are traits of degree, polygenic, they are highly influenced by environment, and they cannot be studied exactly as Mendelian ratios, but they have to study as frequency distributions and also using statistical methods. So this QTL mapping is more to do with those statistical methods. That is what, like, so far you have gained a lot of vocabulary. In this lecture, we are going to learn some, we are going to revise some grammar. So that is the idea of this. And one important question which stands out when we talk about marker trait associations is that we know the molecular markers are qualitative in nature. Okay, they are either presence, absence, or adenine in place of guanine, or a transversion adenine in place of a cytosine or a thymine. So it is always a qualitative one, but whereas your traits, it is a quantitative one. Now the challenge that is in front of all of us is to find the relationship between a qualitative attribute with a quantitative attribute. So then you get into certain problems. But before that, I just want to remind you that mapping populations, all genetics is through the study of inheritance. And for that, from time immemorial, from Mendel's time, we always discuss a biparental crosses. So that is parent 1 with parent 2, and most of the first generation mapping populations, whether it is recombinant inbred lines or F2 mapping populations or back cross or DH, whatever, are advanced intercross lines, all of them are a cross of only two parents. So when we say two parents, we know they are if they are segregating for something, then they have two different alleles in that population. But nowadays in the second generation, like magic, nam and all, we have more parents, so we may have more than two alleles segregating in a population. Now, but as I told you, it is very important now to understand, because the markers are qualitative but your trait is quantitative. So we need to recall how we can represent even a qualitative aspect in terms of quantitative aspect. So I would like to reintroduce you to the biometrician's perspective of looking at a biallelic locus, which is a simple if the alleles are capital A and small a at a locus, you can have three genotypes, capital A, small a, and capital A, small a. And if the frequency of a capital A is small p and the frequency of small a is q, then the genotype frequencies will turn into p squared into 2pq and q squared. This all of you, this probably is your first lecture on population genetics. All of you know this. And we also keep in mind that it is the gametes that come to the next generation and not the parents. So this is always, we explain this in a random mating population where P and Q are rarely equal. But in case of mapping populations, we are at an advantage. P and Q are always equal. P is equal to Q is equal to 0.5. Now, how do we denote the genotypes? If we denote the genotypes on a measurement scale, then we can say, let us say small a is at a, we can denote it at a point, and capital A. And then if we can take the mean of a small a and capital A, it will come this origin point. From this origin point, always small a and capital A are equidistant. Now the question is, so this is called the additive effect. The distance from the mean to the small a or the distance from the mean to the capital A. Only the direction is different, so it is a vector quantity. The direction is different. Otherwise, by magnitude, they are equal. So this is what we call the additive effect. And when you combine the additive effects, you get the breeding value. So then now, how about the heterozygote? How is the heterozygote going to behave? If the heterozygote falls exactly at the mean, we call there is no dominance. If the heterozygote falls in between the 0 point, that is the origin point, and between any one of the genotypes, we call it partial dominance. If it falls exactly at the capital A or small a, we call it complete dominance. If it falls beyond these points in the scale, then we call it over dominance. This is very important to understand this for a later understanding. So that's why I have spent time on this. Now, let us say, how do we calculate the mean of any population? So if you, I have given an example here. Let us say the parent one value of leaf width is 25 centimeter and the small a is 7 centimeter, and the heterozygote, that is F1, after crossing you obtained, gave you 18 centimeter. And you know, in the F2, the frequencies are supposed to be 0.25 and 0.25 for the homozygotes and 0.5 for the heterozygote. And in biometrical terms, you cannot put value 25 as A. It is always the mean. Now, mean is what? 25 plus 7 divided by 2, 16. So how much the capital A differed from 16 is by plus 9 and small a by minus 9. So this is your additive value. As I said, told you, the direction is different, but the magnitude is the same. Coming to the dominance value, it is 18. So it is a little, it is a partial dominance case. It is a neither 25 nor 16. It is somewhere in between that. So it is a partial dominance. So the value of dominance is 2. So if you have to calculate the F2 population mean, we have to remember our statistics fixed classes where we did sigma fxi, the frequency into the multiplied by the value and sum it over all loci, you got the F2 population mean. So we can say in terms of notations, right mean of a mapping population, F2 mapping population is P squared A plus, this is the notation. If you simplify this, you will get into A into P minus Q plus 2 PQD. But this is for only one locus. If we have multiple loci, the real attribute of a quantitative trait came because of polygenes, but the 25, 19, 7 came because of many genes, not because of only one gene. So it has to be represented as summation of A into P minus Q plus 2 into summation of PQD. That is, this is over multiple loci. Now our exact question is that how to partition it to each locus. How do we say this locus is giving this much effect, the other locus is giving the other effect? So this is what we need to explain. So here I have given the same thing again. So this is the sigma fxi. So yeah, all of you may be knowing the regression equations. Regression equations are the crux of the ANOVA also. So if you, and also whenever we measure quantitative traits, we measure it across locations. Dr. Bharadwaj said you may get in Delhi many QTLs, and then again Cuttack multiple QTLs, and so on and so forth. So this is because of the environmental effect. So we need to one thing is that our method has to be precise, and our populations have to be measured at multiple locations. So you will get a different error factors for each environment, along with one y1, y2, y3, that is the mean. So then the genotypic frequencies you already know. We always express everything from the deviation from mean. So that also you already know, A minus mu, D minus mu, minus A minus mu. So this is the alpha is nothing but your breeding value. Please remember, it is the gamete that gets to the progeny. So there are whichever quantitative trait we are talking about, there are going to be multiple genes. All these alleles are going to the progeny, and it is coming because of multiple loci. Here we are trying to look at one locus at a time, and even at one locus at a time, it is the gamete that goes to the progeny. Breeding values can never be measured. They can always only be estimated, because it is the effect of allele 1 and allele 2 combined together that gives the breeding value. So then the dominance deviation is whatever is the difference between the breeding value and the population mean. But so far we have been talking about only in the first order statistics, but there is another important term. But even when we talk about ANOVA also, it is a second order statistics. So we have to talk in variance terms. Now, because the quantitative traits can be explained not only based on means, but also on variance and covariance. So now we are going to talk about we have quantitative trait on one side and marker on the other side. Now how they are going to co-vary is the covariance. That is, let us say covariance X is the marker and Y is the trait. So how they are going to co-vary is what we are going to talk about. So then when we talk about covariance, we have two important measures coming from covariance. One is correlation. We rarely use it in case of QTL mapping, but we do use it a lot in case of Jivas. And in case of regression, we, of course, many regression models are based on many QTL models are based on regression, and even the basic fundamental point of the genomic selection models are also regression, but then they are built upon because there is a problem. You have a huge number of markers, lakhs of markers on one side, and only 300 or 1000 individuals phenotype data on the other side. So when we have to do this, a simple regression equation is not sufficient. We have to go for sophisticated regression programs. It is regression is a very, very important measure. All of you need to know as plant breeding students. So regression, the difference between regression and correlation is that both of them need covariance, but it is the how both the factors co-vary is because in the denominator, if you see, you have the sigma x, and how did what is the dispersion of the variable x and what is the dispersion of the variable y. Both are in the equation, but in case of regression, there is a dependent and independent variable. The dependent variable is always the markers. The dependent variable is always your phenotype in question, for which you are trying to identify the QTLs. Okay, and your independent variables are your markers. Okay, so please remember this. This is going to be very useful later. So then coming on to again, I try to remind you that we are now trying to identify the marker trait associations. So yeah, in simplified terms, we are trying to identify which is the locus that is are the a set of loci, the are a genomic region that is controlling it, right? And we are trying to do this based on means, variance, and covariance. So if there is in simplest terms, let us say a marker is co-segregating with a QTL, then the marker loci, if we calculate the means of these different marker classes, then the marker classes means will be different if they are co-segregating with a trait. So this is what we called linkage disequilibrium or linkage. Both are one and the same. If there, because if there is an equilibrium, then the QTL and the marker are not at all linked, because they will segregate independent of each other. But if there is linkage disequilibrium, then they are going to co-segregate. Then the covariance will be significant. Then because of that, your marker class means will also be significant. So in other words, the parental and recombinance means will also be significant. So you have a QTL in the vicinity. Now introducing you to that, but this is not at all a new concept. This is a very old concept. First explained by Sachs in 1923. So before talking about his experiment, here I would like to just give you a background of the material of that experiment. It is about seed color and seed weight in Phaseolus vulgaris. When it comes to seed color, there are 4 different types. One is the improved yellow eye, where there is a hilum in all pulse crops. In this, if you look at this hilum, the 1/3 of the area has a very strong solid yellow pigmentation. Yeah, it has surrounding this hilum. So this is the improved yellow eye type. And the dot eye, it also has the yellow color, but that pigmentation is very, very small around the hilum. In case of white, they are non-pigmented types, and in nature we have lot of white types rather than the pigmented types. And invariably only in wild we have lot of colored types. And in case of cultivated types, most of them come from white. Most of them are white seeded only. And that's why in white seeded you have a large number of pure lines available, and invariably the white seeded ones are always smaller in size. And there is one more color, mottle. Here it is with like brown, purple, multiple pigmentations are there on this entire seed coat. So this is with respect to seed color differentiations and Phaseolus. Now coming to the genetic determinants of seed color, what is known by 1923 is that it is controlled by multiple components. Some components defined by Sachs are, one is the P capital. This is the pigmentation gene. Later in 2006, this was identified as the basic helix loop helix transcription factor of clade B of subclass 3F. This is basically a transcription factor which controls pigmentation. And then there are a pair of linked genes which are Y and Z on the same chromosome, and that is they are linked. So they are the ones which lead to mottling. Then there is one more gene called extension factor. They named it T. The extension factor means basically there are lots of colors like brown, purple, and other colors. So these genes are called T. But there are though Sachs talks about only one T, there are multiple T actually. And recessive of T always led to a dot type. Now, what exactly Sachs did, in the entire Sachs paper, you will see only 2 tables. The first table talks about the genetic segregation ratios of the 4 crosses I have shown here. And then here I am trying to show you the differences in weight, of these classes. See, in all the female parents have colored parents, improved yellow eye 1310 into male parent is the white 1333. I told you there are several pure lines available. In this experiment, 2 pure lines have been used coming from completely different pedigree. One is white 1333, the another is white 1228. Okay, yeah, in case of the other 3 crosses, white 1228 has been used, and you compare the weight of these. See, all the time the colored ones are much heavier in size compared to the white ones, which are much what to say, lighter in size. Okay, and if you look at the F2 segregates, though we crossed only the improved yellow eye or dot eye with the white, when we looked at the segregants, invariably you got 4 kinds of segregants. You got a white one, which is expected, but the colored ones segregated into multiple ones like mottled, self, and eyed. And if you look at the, I have not shown you the table one. The paper is freely available. You can have a look when you have time. You will always see equal number of mottled and self classes. So that means this is the when it segregates, it becomes it becomes either mottled or it becomes self, because when he used 2 backgrounds, they were in trans linkage, they were coming from different backgrounds. So that is why you had equal number of them all the time. And also you see their seed weights are also almost the same. But if you look at the eyed ones and the white ones, you can see a distinct difference in their seed weights. Always remember, when you look at parents, there will be a huge difference in the parents. But when we look at through the eyes of your marker, here the marker is the pigmentation, that huge differences are not here. You have got very small differences because this is that particular gene that is lying next to the pigmentation or linked to the pigmentation locus that is bringing you this difference. So this is an important thing we need to understand. So now we need to calculate everything based on this F2 data, not based on the parental data. So if you look at the marker class means of this, you will see that a capital PP mean is 31.3, the small PP mean is 26.4, the capital P small p mean is 28.7. Now based on your model, the additive effect and the dominance effect can be calculated using the formula I explained a little ago. You will be getting the slides even at your leisure. You can have a look at it. It is only the parents, parent 1 minus parent 2, but mind you, this has come from the F2 data, not from the parental data. So then you get the additive effect. So this is the additive effect due to that particular locus, which is linked to P. That is why you get 2.45 centigrams. Now dominance effect is minus 0.15 centigrams using the same methodology. So now the crux, as I told you, it is if we have these kinds of pigmentation or some easy to score markers, it is possible to follow a quantitative trait and also measure how much genetic gain can be obtained by doing a marker assisted breeding using this particular pigmentation trait, which is a qualitative in nature, okay, which is very easy to follow also. So this is what Sachs demonstrated. Some of the terminologies I am using may not be exactly from Sachs, but even the additive effect, dominance effect calculations are not exactly from Sachs, but I have derived them for the understanding. So this is the first demonstration between a marker and a QT. So I would like to remind you, see, this understanding of a quantitative trait can be followed using a qualitative trait is there even in 1920s. Okay, so very beautifully demonstrated by Sachs. Now moving on to, so this kind of work was not exactly followed, but rather a different kind of work was followed in Drosophila by a group of scientists. One of the major people in that group are Breeze and Mather. I am not going to here talk about Breeze and Mather work because their idea is to do selections, but here I am going to talk about Tode's work, but today's work is nothing but they are all the same group. It is a kind of continuation of or extension of Mather and Bree's work only. So what did he do? What is that different he did from others is that if you all of you get a chance, you must read the 1961 paper by Tode, because the introduction is very, very beautifully written. What he says is that, see, we all know quantitative traits. We all know they are controlled by large number of genes. We all know they need more statistics to handle them. But that doesn't mean that we immediately give them up, like searching the location of these genes. It is a duty of every geneticist to pursue this and identify the location of these polygenes. This is what he kind of charges the geneticist with this statements, like it is a duty. We are duty bound to do this. So you're using the limited facilities available in those times, because you see that it is only 8 years since we know DNA is the genetic material. The recombinant DNA technology is not born. We don't know even restriction enzymes. They came in 1970s. So in that time, so everything we have to depend on genetics only. So what he suggests is that whenever you have a metric trait, okay, anything is a metric trait. Here the example is the Kete number or Bristle number in Drosophila abdominal Kete number, or Bristle, the leg Bristle number, or something like that. So let us say you have a stock with a high metric trait, and you have another stock with or inbred strain with low metric value of the metric trait, and now the duty of the geneticist is to develop a large number of stocks which have a large number of recessive alleles. Like we always try to develop stocks in such a way that the low value metric character giving inbred strains have a large number of recessive markers along with them. So if we do that, then we have populated a large number of stocks with low value traits, as well as the recessive markers, which can be easily followed in genetics segregation, like generations, F1, F2, and so and further generations and so on. So now let us imagine a high value line and a low value line. The low value line has a recessive marker A and B. It has a large number of markers, and we have identified it in such a way that this A and B, your L, is lying in between the visible markers A and B. Now, if you can test across this line with the homozygous L line, then it will be possible, just based on the phenotype classes, it will be possible to look at identify which are all the parents, which are all the recombinants. The parental classes will always be like all small ab will have low value, and all plus plus will have high value. They are the parental classes. But in case of recombinants, a proportion of plus B will be high value and the rest will be L, and especially in the other case, reciprocal cross proportions of A plus will be low value and all the rest will be high value. So you can easily get parents and also recombinants, and the recombinants also can be further classified into the high value recombinants and the low value recombinants in the two recombinant classes, plus B classes and A plus classes. So but this will be there will be only these many classes if we are talking about only one gene or only one QTL lying between this A and B. If there are multiple genes lying in this QTL, then we will have additional recombinant classes, but they will be very difficult to distinguish in the F2 generation. We have to go for progeny testing to actually unequivocally distinguish them. So now this is nothing but the interval mapping or the use of flanking markers, which we know in later the terminologies which we use when we do in marker assisted selection. We try to go look for whether we have flanking markers, especially in case of if we have to transfer a QTL, we need to look for the flanking markers as well as a peak marker. So this is what in 1961 itself Tode described in his paper. So by the how he concludes his paper is that the main practical limitation of the technique seems to be the availability of suitable markers and the time that can be given to the considerable work involved, because we have to develop stocks that takes a lot and lot of time, right? So this is the so then we have to identify those many recessive alleles, so which is actually very tough job. This is the only limitation, but this limitation was easily solved by molecular markers with this is the kind of the genetic map we had with morphological markers in case of Drosophila, but this is the kind of high resolution linkage map we have nowadays with the kind of molecular SNP markers we have in today's time. So now the limitation that Tode talked about in terms of the availability of marker has disappeared after 2000 and completely after 2010. So the concept of molecular markers, the concept of mapping populations, and the concept of genetic linkage made mapping or tagging of traits a reality, and more so for a quantitative traits. So nowadays you can see a large number of papers across crops where high density SNP linkage maps, even in a crop like pigeon pea, where we have comparatively very low genetic diversity is available, such are developed. And in case of rice and other crops, we have, you saw in the morning, heard about a 50k SNP array, 62k SNP array, not only in crops like rice, but also in crops like pigeon pea, and also in mango, jute, and in all those crops you heard of those things. So now markers are a reality. That is no more limitation, and this is the abundance of markers has the essential one which has moved the plant breeding and the identification of genes and the QTLs towards jivas in case of plants, because jivas is a very old technique when it comes to human genetics, but this became an important technique only because of the abundance of marker in case of plants. And genomic selection, it is only the markers that have made it possible, nothing else. Even if segregation distortion is there in the markers, because we do expect 1:2:1 ratio for any qualitative attribute in F2 mapping population when it is a codominant in nature, but sometimes it may not happen, but even then it is possible to use the QTLs. And why these QTLs are hyped, why should anybody bother about QTLs? You see a large number of examples in the morning. Here this slide you also saw in the morning from Dr. N.K. Singh sir. See, it is just one QTL submergence, but it has been moved to so many varieties, Samba Mashuri, like IR64, IR42, and Swarna, and so many other lines. So because of that, you have the yields have increased from nearly 1.35 tons per hectare to 3.5 tons per hectare. This is the kind of improvement we can the identification of QTLs can offer to the real crop improvement. Okay, so this slide was also comparatively in a different way presented by Dr. N.K. Singh. See, it is only FR13A which was moved into not only just India, but you see the countries Indonesia, Bangladesh, Philippines, Lao PDR, so many countries. And in case of the other QTLs like sub-1 and QBPH12, they were moved to Thailand also, and sub-1A donors from ERI again moved to Vietnam, and the Saltal QTLs moved to Bangladesh, Vietnam, as well as the Indian. And also there is one more QTL which is a very important reproductive stage salinity QTL, which is being mobilized to these many varieties in our country under the leadership of Dr. N.K. Singh sir. And then sir also talked about the drought tolerant QTLs. These are all the different donors of the drought tolerant QTLs. So you saw like the different combinations of these QTLs have been moved into many varieties. And now I again would like to come back to the like the crux of QTL mapping and try to share with you a few important ideas. In the initial time when we identified that monogenic molecular markers for identification of linkage with a QT, see the marker is one allele is small a, the other allele is small b. So though these notations have been used in paper, it is better to use notations as capital A small a or A1 A2, because when you say AB, it gives an idea that we are talking about two different loci, but no, here we are talking about only one marker locus which has two different alleles A and B. And then a QTL, this is also a single locus which has QTL A is one allele, the QTL B is another allele of that QTL. And if they are really, if we cross them, you will get a F1, okay, and then in the F2 they will segregate. You will have the parental classes and the recombinant classes. This is the phenotype means of the subgroups, like these are all the different individuals of the F2 progeny. And then all the progeny, let us say hundreds of progeny, they can be grouped into three groups where they are marker small a as well as QTL marker A and QTL A, marker B and QTL B, which are the parental types and also the F1 type. Whenever we talk about mapping populations, these kinds of ideas do help one. Like we can talk, instead of saying homozygote, heterozygote and all those stuff, we can say like parent 1 type, parent 2 type and the F1 type, because this refers to one homozygote, this refers to the another high homozygote, and this refers to the nothing but the heterozygote. Okay, if these means are significantly different, then we can say yes, the QTL is linked with a marker. So now you take any polymorphic marker between parents A and B and see how they are segregating in the F2 mapping population or any mapping population for that matter you have, and then go and calculate the marker class means, like what is the marker A mean, what is the marker B mean or parent 2 mean, and then what is the F1 mean, that is the heterozygote marker class mean. If they are significantly different, then you have caught hold of a QTL. Now this is the same, this is akin to what Sachs said. Now we move on to what Tode said. Here we have two markers, okay, marker 1 and marker 2. Here also, like the AB, we have used the same notations AB. Like there is a marker 1 and marker 2, and your QTL is in between these two markers. This is what exactly Tode envisages, but with molecular, rather than molecular markers, but with recessive markers, because recessive markers means they can unequivocally followed. Their crossing over events can be followed very easily. That's why we need recessive markers to be along with the low value QTL. But in case of, if you have molecular markers, such a the cis trans both linkages are perfectly fine with us. If it is a dominant marker, if it is a co-dominant marker, absolutely no problem. You know all the three classes very distinct from each other. See, this is more with from in terms of like what happens when you use a flanking two markers instead of one marker. This talks about selection efficiency, but here the idea is not selection efficiency. This is to bracket your QTL within this marker. So this is the idea. So now this is the same thing. Now I would like to give you an example. Let us say this is the trite. Okay, this I have taken from Falconer. Okay, so this is your molecular marker, capital P, capital P, capital P, small p, or small p, small p. You can treat it as a pigmentation marker also. It doesn't matter. You can follow it in the population, and it is a qualitative trait. That is the only criteria. But now there are multiple scenarios you will be having with respect to QT. The QT may be what to say, came from the parent, both the parents, capital P, capital Q gametes came together, or from one parent, capital P, capital Q gamete came, but from the other parent, capital P, small q gamete came, or from both the parents, capital P, small q gametes came, because see, it is only a linked marker. So recombination can happen between the marker and the your quantitative trait. So because of this, actually this is invisible. The QT is invisible. You are imagining that capital P is with capital Q, but actually by the time you score the F2, in F1 meiosis, crossing over would have happened. So you will have some individuals of capital PP which are capital QQ, small q also, small q also. So then, but you can let us say, you can say there like proportions, recombination is R, then you can say parental fraction is 1 minus R, but you know there are actually 4 gametes. So we have to divide the recombinant fraction to represent like a capital A, like a capital capital Q, capital P gamete, capital Q like small p gamete, similarly small q capital B gamete, and small q gamete. So there are 4 gametes. So you have 2 are parental types, 2 are recombinant types. So you have to say the recombinant fraction is R, but since there are 2 types in each, we have to say R by 2 is the class, each gamete class, and similarly parental fraction is 1 minus R by 2. So then I have done is only represented 1 minus R by 2 into 1 minus R by 2 gives the frequency of this. So like this I have worked out the frequency of this, and the value is always depending upon the QT class. You can write the value. If the QT class is capital Q, it will be A, and heterozygote it is D, and the other parent minus A. This is very simple, but you are remember, you are not following this frequency and this value. All you are following is only the marker. So the marker class frequency is 1 by 4, 1 by 2, 1 by 4. The entire class is actually condensed or confounded into the marker class. So if you write them in notations, you will see the A is nothing but your effect, and R is nothing but your recombination fraction. They are within the same notation. You can never remove or separate the R and A in this case. When you have a single marker and they're trying to follow the QT using that single marker, the recombination frequency, this is only theoretical. Okay, in reality you will never be able to identify this. You have to follow only the marker class. So this is one of the demerits of the single marker analysis also. Whatever recombinations happen between the marker and the QT, that will never be able to be traced, and because of that your values will always be inflated, and you will always find a large number of false positives. So if you in theoretical notations, if you calculate all this, you will see the marker mean of each class is 0.5 D, and if there is no linkage, that means they are independently asserting the QTL and the marker, they are not at all linked. But once they are linked, you will see that their marker class means are different. So you will get a so you can now easily reject the null hypothesis. There is only one more thing I want to introduce. This is how a single marker analysis is done. But if we have to move towards the interval mapping or the simple interval mapping, we need to understand 2 more concepts. One is the regression, which all of you I believe already know. So here I will try to talk about conditional probabilities, because the conditional probabilities are essential to identify the maximum likelihood estimates. See, for example, you let us forget QTL for a moment. You are constructing a genetic linkage map. When you are constructing a genetic linkage map, all you have is the data on the genotype data of the entire mapping population across multiple markers. You don't know whether they are linked with one another. In this case also, we are estimating the maximum likelihood estimate, whether if they are linked, what would be the conditional probability. If they are not linked, what would be the conditional probability. And then based on that, we get a maximum likelihood estimates. If the and we get we imagine, let us say, if the recombination distance between these 2 marker 1 and marker 2 is 0.1, so 10 centimargan or 20 centimargan, 30 centimargan, 40 centimargan. If is the moment is a 50 centimargan, You are saying that they are independently asserting. So we try to see what is the different maximum likelihood estimate for each of these different centimargins, and wherever you are getting a maximum likelihood estimate value, we say yes, this is the distance at which these two markers are linked. Let us say you get the maximum value at 20 centimarkers, then you say yes, this is the distance between these two markers is 20 centimarkers. Now let us move on to the next marker. So this is how we can generate even genetic linkage maps. The same concept here. One slight difference is that I am in case of all of you might have learned the conditional probability in your younger classes also. See, you are always seeing the marker classes. So marker class is always the given probability. So it is your marker class probabilities. We are seeing the data, but your QTL is not. You don't know whether it is there or not. So you have to estimate it. So the idea is that probability of the QTL present there, given the marker, is already this particular one. Okay, so then the formula is that probability of QKMJ divided by probability of MJ. So this is how we have to, but how to calculate the frequency of MQ and this different gametes. I have already told you that marker Q gametes, they are non-recombinant fractions. So it is 1 minus R by 2, and recombinant fractions are R by 2. Now you can replace probability of MMQQ is nothing but probability of MQ into MQ. So 1 minus R by 2 into 1 minus R by 2. So like this, you can write the probability of all the three classes, and then you can add up all these classes. Then you can write the conditional probability, because PR of MMQQ, which you have already calculated, 1 minus R squared by 4 divided by probability of marker, which is nothing but 1 by 4, this classes. So because this is nothing but the capital M parent 1 class. So if you divide it, you will end up with 1 minus R squared. See, now you have got the conditional probabilities. So this is possible to work out conditional probabilities for each marker class and each marker locus. And then if you have this conditional probabilities worked out, then you can say based on these conditional probabilities, we can estimate whether the QTLs are there or not by comparing again their maximum likelihood estimates. So now, but the interval mapping is little more complex than this, because see, in the interval mapping, you know only marker 1 and marker 2. You don't know whether QTL is present there. Even if it is present there, you don't know exactly where in this QTL interval your QTL is present. So now we have a little more complex situation, and also we also know the genetic distance between marker 1 and marker 2, but that is C. It is measured by the fraction C, but or you can say R also. Any notation can be used, but there is a distance between marker 1 and the QT, and also there is a genetic distance between marker the QT and the marker 2, which are represented by C1 and C2. So instead of writing the classes for MMQQ, now we have to write the probability for 3 genes at a time. Considering 3 genes at a time, we have to write the probabilities. So now we have to, like as I told you, in case of genetic linkage map construction, you obtain the estimates saying that like 5 centimargin, 10 centimargin, what could be the estimate. Similarly, here also we say my QTL is M1 itself, my QTL is 2 centimargin away, 4 centimargin away, or 6 centimargin away. Like this, we do a scan between M1 M2. But please please remember, in case of a single marker analysis, it is only one locus at a time compared with the QT. Similarly, here it is only one interval at a time compared with the quantitative trait, are associated with a quantitative trait. So then this is simple interval mapping, but this also has some problem, because simultaneously the population is also segregating for QTLs on the other chromosomes. The effect that is confounded with is always confounded with the QTL effect which you are measuring. So that's why people moved on to composite interval mapping. In case of composite interval mapping, we are trying to use a regression approach where the first thing is that we have to first do a simple interval mapping and identify which are all the different like markers that are associated with a QT, which are significantly associated with a QT, and then we have to say, okay, these are all the multiple markers that are associated with a QT. Now use these markers and let us try to build a model. In this model, instead of your the marker that you are testing is the factor, but the other markers are used as a cofactor in the equation, so that the effect coming from each marker can be separated. Okay, so in case of inclusive in case of composite interval mapping, we have to keep on trying with a large all the like significant markers. But in case of inclusive mapping, we are building a composite linear regression model of all the marker effects at the current testing position. So here we can estimate the QTL effect as separate from the background effects. And but then there is other models also where we can like multiple interval mapping. In case of multiple interval mapping, we can even like, let us say the QTL1 is interacting with the QTL2, that can also be put into a genetic model. So in case like I before winding up my lecture, I would like to give you a few key points. Generally students confuse the effect and the variance. Effect is always a first order statistics. Whatever you saw, A, D, minus A, additive and dominance effect, these are effects. It should not be confused with phenotype variance explained. And phenotype variance explained is because of variance term. This is not a that is a second order statistics. In case of effect of alleles, what you mean is that if I like use this particular marker for in my marker assisted selection, that A unit of gain, that is capital A minus A, whatever the effect of that allele, I will get if I replace one allele with the another allele. But in case of variance term, we are talking something completely different. See, we are saying this parent 1 and parent 2 have this much difference for the quantitative trait in terms of value. But when you look at the F2 dispersion, that is the variance, how the completely the F2 population is segregating from transgressive segregants to including the parental types to the next is transgressive segregants, that the entire spectrum of variation, how much is this? So the spectrum of variation is the variance. So in this variance, what is the proportion of variance that is being explained by this particular activity? So that is what the R square or the coefficient of determination or PVE talks about. We should not confuse these two terms. And the large score is most of you know, because large score is as a genetic students, we learn it in two contexts. One is like whether the genes are really linked or whether the QT and the gene are linked. If the we always compare a null, we always have a null hypothesis. The null hypothesis says that the genes are independently assorting, or the QT and the gene are independently assorting. And but then we estimate the conditional probability for that particular case that they are independently assorting. Then we estimate conditional probabilities for different scenario of genetic distance between the QT and the marker. And then we again get a maximum likelihood estimate, and then we compare these two, and then we obtain the ratio of these two is called the large score. And in this to avoid the type 1 error or the false positive, we try to go for a very high large score. If you look at papers, you might have seen traditionally the QTL papers would use a large score of 3 or so, but if you move on to GWAS papers, you will see a very high large score or very high p values of 7, 8 you'll be using. That is because the number of markers, whenever you increase the number of markers, and because generally we don't go beyond a specific number of 200, 300 per population size. Very rarely we go for 1000 and more than that. But we keep our ability to increase the number of markers has increased multifold. So because of this, the moment you increase the number of markers, you should even be careful and go for the maximum number of like a high value of large scores to be like certain that really you are following a QTL genomic region. And another important thing you need to understand is the confidence interval. That is, what is the confidence interval your QTL is lying in this case. So confidence interval can be calculated by two methods. One is the 530 divided by NP, where N is the population size and P is the percentage of PBE expanded by the QTL. But we don't calculate it there, that way. None of the algorithms you use like QTL cartographer or QTL and all of them, they rather do a bootstrapping technique to get the confidence intervals. That is means redoing the analysis by resampling your data and then trying to ascertain whether it is really this. And this is how the just a second. Yeah, this is just to tell you like whether how to calculate the genetics distance between marker 1 and marker 2 is the C12, which is nothing but a component of the genetic distance between marker 1 and QT and marker 2 and QT. So this is, there will be 9 types of gametes and 27 genotypes in this case, because this is a three loci case. There is marker 1 and there is a marker 2 and there is a QTL also. So you will have to calculate those many things to identify those many computations have to be made for each locus and also for within a interval for each point we are estimating at the genomic scan you are doing. And then moving on to this is you can fit the entire model into the equation where what is the distribution of marker 1 class and what is the distribution of marker 2 class and what is the genetic effect. And see in this equation you can see the A and R can be separated. So this is this has been written only to show you the R, nothing but the fraction can be separated from the effect. So this is where your interval mapping scores over your single marker analysis, where A and R terms could not be separated, but here it is possible to separate these terms. And this is how the estimated QTL location with the confidence interval looks like. This is the confidence interval, this is the peak marker, this is the linear chromosome, because eukaryotic chromosomes are always linear. So we represent it like this. So with this I conclude my lecture, and my sincere thanks are to Dr. K.V. Prabhu, who initiated me indirectly into this, and then Dr. N.K. Singh and Dr. Mahapatra sir, who directly initiated me into this, and also all the RAs, SRFs and IRA students who are working in various capacity in this, like various programs in which I am involved in this. Thank you one and all, and thanks for your patient listening.
Thank you, madam, for your nice presentation. Now there are few questions.
Excuse me, ma'am.
Yes.
Ma'am, can you answer some of the questions?
Yeah. Which is the best method? The best method we would say is the composite interval mapping. It's better to use, but because composite interval mapping as such first does simple interval mapping and then only it moves on to composite interval mapping. But it is always better to and here I would like to quote Bharat sir, it is the phenotype that is very important. Okay, your phenotype measurements have to, because generally with the marker data we do minimal mistakes, especially in the SNP era. The chances for mistakes have come down, many things are automated, and once you take the identity of your samples correctly, then there is hardly any chance for mistakes in terms of marker data. But lots of mistakes can come from your phenotype data. So that is why the single so it is better to go for composite interval mapping. This see, the single interval mapping and simple interval mapping are one and the same. It is like only looking at one interval at a time. Okay, we are not at, though we have a genetic linkage map, we have a large number of markers. We are not calculating everything at a time. We just take one interval and scan that interval and say, okay, in this interval QTL is there or it is not there. If it is there, it is at this particular location. This is what single interval mapping does. It dissociates the recombination fraction from the effect. Okay, but in case of composite interval mapping, it also takes into account whether the whatever value you are getting because of this particular QTL interval, is it owing to only this QTL interval, or there is influence of the linked marker also. So that is why many times you will see in composite interval mapping, you will give the nearby markers, 3 markers or 6 markers or 4 markers on either side as cofactors. That is the difference between the simple interval and the composite interval mapping. Direct molecule associated with a trait can be used to directly. You can try, but it will not work many times, because see, there should be a microcollinearity because in genomics another important area is comparative genomics. Unless and otherwise there is a microcollinearity, that means your species are so much co-evolved and just now diverged a few thousand years ago, then you can like expect that. But otherwise I would advise better not to do that. Okay, if you know the gene, now let us say it is the BHLX H helix marker, you know, or it is the sub1A gene, you know, or it is the in case of pistil, you know, the gene. So if you use that particular gene, that will work. But the linked marker, no, it's very rare. It's better not to. Magic population, yes, you can use magic population in both self-pollinated crops and cross-pollinated crops. There is absolutely no problem. And we develop magic only to identify QTLs only. So it can be done. And in case of magic population, the good thing is that in addition to your the additive effects and dominance effect, you can even identify interaction effects. That is, we call epistasis and genetics, that can also be identified using magic populations. It's equally amenable to both self and cross-pollinated crops. The good thing is that in magic you are sampling so many alleles, but in case of biparental mapping you are sampling only 2 alleles at a time for the locus. But in case of magic, from 2 to 8 alleles you can sample at a time. Okay, so that is the beauty of magic population, because the genetic diversity of the base you have extended.
Thank you, madam, for nice presentations. And now there is break for lunch.
Thank you for the opportunity, sir. Thank you, one and all. Thank you for your patient listening also. Yeah, thank you. Yeah.
Hello. Hello. The session will be continued from 3 o'clock. Rejoin.
All participants are requested to be present at 3 o'clock for other 2 lectures, which will be also be very useful for the purpose of research as well as for students' benefit also. So please be back at 3 o'clock. Thank you.
अभी ये मैं एंड कर रहा हूँ ब्रॉडकास्ट एंड कर रहा हूँ सर ऑनलाइन